In terms of why MTP Connect is interested in clinical trials, I'm not going to read these out for you, but you can see that one of the 7 strategic priorities for MTP Connect as a growth centre is around strengthening Australia as an attractive clinical trials research destination. Why do we want to do that? There are a number of reasons. I'm going to come back to that in a minute. What we've been able to do with our funding since 2015 has been probably more active than a lot of people are aware. And this is some of the examples of some of the programs that we've been able to support since 2015 with that growth centre funding that we received. So we've helped set up the Garvan molecular profiling platform, the Cyro protein production platform that was recently launched. We helped set up the Ab initio facility across the road, and Paul's going to talk later, which is very, very close to receiving TGA license, which is fantastic to see. And then there's been a number of other programs, including the Australian Cardiovascular Alliance, working with them to set up their device collaboration, CTIQ, and a number of other things that we've done to help build and make more accessible clinical trials. So why do we want to fund clinical trials? I think the first reason is that clinical trials increase the speed of Australian patients accessing medical innovations. I want to highlight this as a case study, and this was a study that was published in Nature earlier this year. This is the second patient to receive what was then the investigative treatment known as CAR T-cell back in 2012. And the study that was published in Nature earlier this year was a follow-up to see where he was at. 10 years later, he is essentially considered cured of the cancer that he had, that he was treated for, and he still has circulating the cells that will kill cancer. And this is the kind of thing that we want to see. Clinical trials can and do save lives. Sorry, just going to close it for a second. Sorry about that. So this is the report that we produced on clinical trials last year, May 2021, to align with Clinical Trials Day last year. It was an update to the report that we did in 2017. If you're interested in clinical trials in Australia, it gives a really solid background. And the 2021 update spends a bit more time talking about where we've come to and where we should go from here. I used to be a little bit apologetic talking about the economic contribution of trials, particularly when talking to academic kind of audiences. But if you want to change politicians' minds, if you want to encourage them to invest in building something, what you need to do is show them the jobs and the money. And part of what we did in this report was that the contribution of Australia's clinical trial sector is enormous. We found that in 2019, expenditure on trials in Australia was 1.4 billion. That was direct spending on trials, so no multipliers added to that. That was an increase of 5% between 2015 and 2019, 5% per annum, I should say. It is a sector that employs 8,000 people, and again, that was 4% year-on-year growth between 2015 and 2019. You'll note that these numbers are all data from 2019. While we did this report last year, 2020 was, for obvious reasons, a bit of a blip. So that's why we're using 2019 data. And between 2015 and 2019, we saw a 7% increase in trials that were started year on year between 2015 and 2019. So that growth is continuing. Our share of global industry-sponsored trials is about 5%, which is significantly higher than our population. So what we're seeing is considerable growth across all of the metrics that we looked at. But the contribution of clinical trials is more than just the amount of jobs and money that gets spent. Yes, there's employment, but also there's a number of different measures that are used, different reported multipliers. I think the largest one is about an 8-time multiplier. We didn't.
You know what I like to measure?
Pick one of those. We merely pointed out there are a number of different ones of these, varying different rigor that have been used to develop them. The highest one is about 8, but they're generally sort of around between 4 to 6 times multiplier. And those benefits come from flow-on benefits like improved treatment outcomes, additional qualities, improved standard of care across the entire health sector, improved research culture and infrastructure, research becoming business as usual in hospitals, hospital staff and researchers building their skills and expertise. So there's a huge contribution that gets made there. This is the data that we produced in our 2022 sector competitiveness plan, and it includes the data from 2021. And so you can see that despite the drop off in 2020, obviously due to COVID, in 2021, we saw that continuation of the 5% year-on-year growth in trials begun in Australia. In terms of where those trials come from, the breakdown by sponsor type, it's still about 2/3 non-industry, 1/3 industry. And in terms of the expenditure, industry is certainly larger, industry-led trials, but non-industry is certainly incredibly important. In terms of the breakdown of growth by phase, perhaps unsurprisingly, it's phase 1 where we've seen the biggest growth. And that's where groups like New South Wales Health, Victoria Health, the other state health groups, Austrade are really focusing on building Australia as a phase 1 destination. And I'm sure we'll hear some more from some of our other speakers about why that is. Interestingly, we've seen phase 3 drop off over that period of time. And I think the other really interesting point there is that we're seeing a very small growth in the number of device trials happening in Australia. And that is an opportunity that we really need to look at. And there's a number of groups like the Australian Cardiovascular Alliance who are trying to take advantage of that and use particularly first-in-human device trials as an area of growth. In terms of Australia's share of global sponsored trials by therapeutic area, oncology is far and away the biggest. I think it's something like about 50% of the industry-sponsored trials that happen in Australia. Over the period 2015 to 2019, we saw an increase in oncology, an increase in respiratory and neurology. We did see a very significant drop-off in infectious disease. As I said, this was before COVID, and so it's unclear where that is at now. We did see a very small increase in ophthalmology, but the overall number of trials there was very low, so hard to know whether that's a statistically significant change. In terms of what we can do to grow the number of trials in Australia, there are a number of impediments and a number of different groups working to address these. Firstly, things around governance and improving the different guidelines state to state, site to site is an area where we can still do a lot of work. Patient recruitment and economics is an area that we need to concentrate on. And I think we need to be aware of the trials that we should be going for, which is not necessarily huge, uncomplicated trials where the ability to enroll large number of patients is key. What we should be doing is leveraging the fact that we have a very high tech, good sort of dedicated infrastructure for trials and be concentrating on complicated, difficult trials, new, different kinds of modalities. Finally, there's a huge gap in terms of the number of people and capacity to do the trials, in particular, clinical trials coordinators and clinical research associates. And that's an area where MTB Connect through our READY program is working hard to increase that. There's also a number of opportunities around building patient awareness, engagement, and patient centricity. In particular, helping people from a non-English speaking background understand the opportunity around clinical trials and helping them become involved in an ethically appropriate way. There's a number of things going on around precision healthcare, and innovative trial design is another area where Australia can leverage our advanced health system to take advantage of that. The priorities for the future include things around efficiency in ethics and governance processes, the ability to streamline applications for ethics so that you can do it in once and it's recognised across multiple states. It's going to be absolutely key. The big feedback that we got, particularly from global pharma, is that when we're trying to get phase 1 trials happening in this country, what we need to do, what we're doing essentially is competing with other countries. The CTN process is a huge advantage, but we also need to be understanding that that ability to get ethics approval quickly is what's key. We need to grow the clinical trials workforce and capabilities, and that is a huge gap at the moment. I'm not sure how many early career people we have listening, but if you're interested in that area, have a look at the MTP Connect's website. There are a number of training opportunities available there. We need to enhance patient recruitment. Australia is never going to have a huge population. We're never going to be able to compete with Eastern Europe or Southeast Asia, but we do still need to be able to access, make aware of the opportunity as many patients as possible. And finally, we need to enhance transparency of the state of the sector. There is still data that would increase our understanding of what we're doing, where and how, that is not easily available. In terms of where to next, I think there are a number of things that we can do in terms of how clinical trials are run in Australia. This was a really interesting publication that was put out. It was either earlier this year. It was earlier this year, with a number of names that people would be probably familiar with. I think the phrase they used, which was really nice, was that consumer engagement in clinical trials is the gold standard, but it's not always what people do. It was a much more pithy way they had of putting it, which is in the notes for this slide, but I can't remember it off the top of my head. Consumer engagement is one sort of clear area. There are a number of ways that trials can be made more friendly for participants. And the other thing I think that is going to be really important is diversity in clinical trials. They're most easily accessible to perhaps educated Anglo people in Australia, and increasing that, one, it increases the ability for people from non-English speaking backgrounds to get access to medical innovation, and two, it increases the population that we can enroll from. Technology in trials is another really interesting area. This was another publication that came out earlier this year talking about standardization of digital endpoints for Alzheimer's disease studies. They reported something like 80 different endpoints that were being used for sleep and various other things related to Alzheimer's disease. This was a consortium of pharmaceutical companies who were coming together to agree what the most useful, relevant digital endpoints were for Alzheimer's studies, so that they could all agree on those, use those, and they were standardized going forward. I think that's going to be a really interesting approach going forward. So that is a very quick overview of what MTP Connect does and the clinical trials landscape in Australia. Those are our contact details. So if you're interested in learning more, you can find our reports online. We have a very active podcast, and you can follow us on social media. So with that, I would like to introduce Judy Bingham, who is our next speaker. Judy is the Executive Director of Easington Product Limited, a product development consultancy established in 2001 to assist Australian biotechnology and medical technology companies bringing innovative products to market. Services include developing regulatory strategy, planning development programs, and managing manufacturing non-clinical and clinical studies. Judy has a broad professional experience in the former role as Senior Director Consultant at Kendall International, now CSL Health, and pharmaceuticals company Organ, now Pfizer and CSL. Judy has a background in pharmacy, and prior to joining the industry with Director of Pharmacy at the Centre, Specialist Priority Research in Melbourne. Judy has a Bachelor of Pharmacy from Monash University, a Fellow of the Science Society Hospital of Pharmacists in Australia, and a Master of University. She also graduated with Chief of Company Directors Formerly a Director of Arts. Over to you, Judy.
Okay, thank you. Can you hear me okay? No.
Are you talking, Judy? We can't hear you.
That's a problem. My sound is on.
I can hear you.
We can't hear you just yet.
Okay, so is it coming through Zoom but not through the main speakers?
It's coming through
Just
Zoom.
talk for us, Judy. Try again.
So it seems to be coming through Zoom but not through the main speakers in the auditorium.
Oh, we got no sound. Yeah,
And I'm certainly not on mute.
it's come through Zoom, just not into the room. Helpful. Just talk for us again, Judy.
Okay, testing. Can you hear me now? No.
Sorry, everyone.
Can people on Zoom hear me?
Yes, loud and clear. Yep.
Yes. Okay, thank you. So it's a problem with the sound in the auditorium.
Sorry, everyone, it's just not coming through in the room, so we're just going to solve that first. Sorry, can you just try one more time?
Okay, testing. Can you hear me now? And I am sharing my screen. I don't know the people on Zoom can see it.
Yes, we can see it. Yep.
Okay.
Yes, I can see it.
But it doesn't look like it's showing up in
Try now.
the auditorium. Can you hear me now?
Try now.
Okay, testing.
Say again.
Okay, try again. Testing.
Try now, Judy.
Okay. So I was just saying I'm sharing my screen.
Sure.
People on Zoom can see it, but I don't know if you can in the auditorium.
She's not muted. Oh, Judy, say something.
Okay, testing. I think you've got me up on your screen now, have you?
We have sound now. The room has helped me quite well. We have some work here, and we've sorted all that out.
So I'm sharing my screen. Can you see it?
Yes, we can. I'm just going to turn you
Okay.
down. All right. When you're ready, Judy. Thank you.
Okay, so we're right to go ahead. Well, after that little technical glitch, thank you very much, everybody, and good afternoon. So I've been asked to speak today about clinical trials in Australia, which is the theme of the seminar. But as was mentioned, I work with small companies assisting with their drug development programs from a clinical and regulatory perspective. So my presentation today is thus focused on clinical trials in drug development. Okay, so I'm going to talk about clinical trials in the context of bringing new medicines to market. And in parallel with the clinical development program, the development of the drug product to use in those clinical trials. And lastly, a little bit about the Australian environment for clinical trials from a regulatory and industry perspective. So I'm sure you've all seen this graph before, as it's one that's commonly presented, but I've included it to emphasise just how important clinical trials are in the development of new medicines from a time, a cost and a risk perspective. So if the average development time for a new medicine from research to market is about 15 years, clinical trials take up about 7 years, so roughly 50% of the total time. But importantly, of all the potential candidates that come out of research labs, only about 1 in 5,000, 1 in 10,000 actually gets to market. So that's a pretty high failure rate. And clinical development is the longest and the most expensive phase, taking on average up to 68% of total development costs. And these are, of course, average figures, and we've seen recent examples of COVID vaccines which have been brought to market in record short time, albeit with huge injection of funds by taking lots of risks and enormous support from the regulatory agencies, from FDA, from EMA and from TGA, but also with large fallout along the way, some of which resulted in the Australian vaccination program being disadvantaged compared with other developed countries. Okay. So with all development programs, we should start with the end in mind. And I think it's something that we don't always think about, but we need to be thinking a long way ahead as to what the product in the marketplace is going to look like. What's the target we're aiming for, so that we can prepare a roadmap of how to get there and plan accordingly. Because if you think about, if you don't know where you want to go, it's pretty hard to get there. So you need to define your target to best you can, and then map the path to reach that target. Of course, we can't make all decisions up front, because the next steps very often depend on results of current studies, and each study builds on the results from the previous study. But as I said, it is important to have a roadmap and follow it as efficiently as we can to avoid meandering programs that cost unnecessary time and money. Okay, so in terms of trials then, we start by thinking about what the product in the marketplace looks like. What are the claims and indications that we're planning for? And what are the population targets? Are we talking about different age groups? We're talking about different severities of diseases. Are we talking about prevention? We're talking about therapy. All sorts of decisions that we need to be planning for. Okay, and then the important question of how we're going to assess and measure if our product actually meets those claims. So each phase of development does build on the earlier phases in a stepwise approach. So phase 3 will build on phase 2, phase 2 will build on phase 1. So we'll just look at those phases in a little bit more detail. So our phase 3 studies, and generally we need 2. There are lots of examples where products have been approved on 1 pivotal study, but the general concept is that you need 2 studies. So they need to be designed, managed and reported such that the dossier that's submitted to the regulatory authorities for approval includes the key data required by the agency to confirm the product's safe and efficacious for use in the population studied in the clinical trials, but as far as possible in the broader population that the product may be used in a post-marketing setting. So what are the study objectives and endpoints, and how will the data be analysed to confirm that the objectives have been met? And sometimes the agencies don't always agree on the endpoints. So you can have a study objective that's agreed, but I have seen agencies, I've seen EMA and FDA request different endpoints and analysis for the same objective in the same study. And indeed, in the study I'm thinking of, we actually had 2 statistical analysis plans and 2 study reports, all coming out of the one study. So it can be challenging to meet global requirements, but I guess that's all part of the planning that we need to be taking those considerations as part of the study design. So in defining the study population, we need to take into account as far as possible a population that might be using the medication in a post-approval broader setting. So as I said, thinking about patients with comorbidities, there's 4 on that slide, but I think, the COVID vaccines were a good example where, because the initial studies were conducted very rapidly to meet, particular objectives, women of childbearing potential weren't considered in those initial studies. And when the vaccines became available, women who were pregnant or planning to become pregnant were left without any good information on which to base a decision as to whether they should receive the vaccine. Now, those studies have subsequently been done, but it was an example where it wasn't done pre-approval, as would routinely be required under a standard development program, if such a thing exists. Okay. And importantly, the size of the safety database is critical, in that you need to have sufficient patients who've been exposed to the drug for a sufficient period of time to demonstrate the safety profile of the product and the adverse event profile. But even with large patient numbers, tens of thousands that were studied with the AZ COVID vaccine, side effects emerged in the post-approval setting that hadn't been identified in the pre-approval studies. So rare side effects can emerge later, and that's why that post-market monitoring is important on an ongoing basis. So we also need to be thinking about the comparator, and I know Craig's going to talk more about how you choose how you manufacture your placebos and so on. But I think in terms of the design of the clinical trials, we need to be thinking about whether we're using a placebo comparator or an active comparator, and each of those have advantages and disadvantages. Placebos can be considered unethical if there's already an acceptable effective treatment available, but active comparators can be risky, especially if the comparator is also a potential competitor. And lastly, because of our pharmaceutical benefits scheme, phase 3 studies usually need to incorporate collection of quality of life data, so that health economics can be done, and the cost effectiveness and cost benefits ratio of the product can be considered in terms of funding submissions. So the data that we need to support the phase 3 program will come from your phase 2 program. So for example, you really need to know at the end of your phase 2 program, you really need to know what the safe and effective dose is, because in phase 3, you do not want to be doing dose finding studies. You need to be as confident as you possibly can be that the dose you take into phase 3 is the dose that you're going to take to market. And similarly, if you're going to use endpoints that the regulatory authorities might consider novel or different from the basis on which similar products received approval, these should be evaluated in your phase 2 studies, so that you've actually generated some data that you can use to present to the agency in support of your design for your phase 3 studies. Okay. But so traditionally phase 1 studies are considered to be first in human studies. But they're not always, of course, and they're not always done in healthy volunteers. So traditionally we did first in human phase 1 studies with a single ascending dose, and then possibly multiple ascending doses in healthy volunteers. But more recently, patient cohorts are being included as well, with the aim of generating either some biomarker data or possibly even some preliminary clinical data, because obviously, companies want to, and potential investors want to understand the potential value of the product if they're going to invest in the more expensive phase 2 studies. But some phase 1 studies may be done much later in the overall development program. So, for example, thorough QT studies which generally are done in healthy volunteers, but not done until during your phase 3 program usually. So although the opening slide shows drug development in a sort of linear fashion, more frequently it's not actually linear, so that you will have ongoing phase 1 and phase 2 studies in parallel with your phase 3 program, and sometimes in parallel with phase 4 in a post-approval setting. But whatever phase of clinical development we're considering, the product used in the trial needs to be appropriate for the stage of development. And I think this is a challenge for quality managers who are responsible for the product development. Firstly, particularly for phase 1 studies and early phase 2 studies, to know what standard is expected at each stage, and also to meet the timelines imposed by the clinical researchers who are ready to start their clinical trials. It's the single biggest factor contributing to delay in the start of clinical trials is in fact delay in availability of the product for the trials. So that's something that the manufacturing team need to plan for, because often with, as with other programs, of course, there's, unintentional delays and so on, but we really, and manufacturing happens behind the scenes. The press releases go out based on, first patient enrolled in the trial or ethics approval or something else related to patient care. Rarely do companies put out press releases saying, well, we manufactured our product. But it is critical that the product is available for the commencement of the trials. I just want to make a comment on the quality standards for phase 1, and I think this is something that is probably warrants some discussion later in the Q&A session. But as we all know, the ICH standards were initially designed for marketed products, and it's not a requirement that early phase products meet always ICH standards. So then the question is, well, what standards should they meet? And we don't have any Australian specific guidelines for early phase products, so we tend to rely on the EMA guideline that I've put up on the slide there. And that is a really useful guideline that I think, Australian companies can use, but it's not a formal requirement in Australia. And the other aspect to that is compliance with GMP. And I think, again, this is clearly open to interpretation, where the regulations state that GMP isn't required for goods prepared for the initial experimental studies in human volunteers. And that's interpreted by TGA as phase 0 and phase 1 trials. But there's a lot of ambiguity, I guess, and different ways to interpret that statement. I think what is important is for phase 1 studies that the formulation, it may well be an early formulation and not necessarily the formulation that you plan to take to market. Can be a simpler formulation, and generally you'll have less stability data, and it can be a simpler presentation. So, you might be planning to take to market a preloaded syringe, but you're not going to manufacture preloaded syringes for your phase 1 study. Most of the phase 1 studies will be done in either phase 1 units or in hospital settings, and so hospital pharmacists will often get involved in, preparing final dose forms for phase 1 studies. But nonetheless, you need to plan and, as far as possible, provide a product that is suitable for use in the study. In my hospital pharmacy days, I remember we had a trial where in the non-clinical studies had estimated the starting dose in the clinical trials to be lower than in fact it was, and we ended up having to use 30 vials to make up each patient dose. That's pretty time consuming for the hospital staff to have to do that. So plan as far as you can to present your dose form in a way that is appropriate for the doses planned in the clinical study. So by the time we get to phase 2, you really should have a product that is reasonably close to the one you plan to take to market. You'll remember there was quite a lot of publicity about the Pfizer COVID vaccine that needed to be stored at -70, and the logistical challenges that caused because hospitals don't routinely have readily available -70 freezers. And so you should be aiming to, by the time you get to phase 2, have a formulation that doesn't require storage at -70 and sufficient stability data that you can prepare, the doses can be prepared in advance. You need to be thinking about your patient population. So if you're doing studies in pediatrics, do you have a dose form that's suitable to administer for children, for example? And similarly, what does your placebo, how are you going to blind the study so that your placebo is visually identical to the active, and if it's an oral formulation, tastes the same, and if it's topical, has the same sort of physical properties and so on. So formulation for your placebo needs careful consideration as well. Thinking about the methods and your product development from a CMC perspective, I have seen examples of products coming into phase 2 that were manufactured at small scale for non-clinical and initial phase 1 studies, but in fact the method wasn't suitable to scale up. And so then you have to go through a whole program to redevelop and change your method, which at phase 2 is feasible, but by the time you get into phase 3, you don't want to be changing your methods then. At the late in development, the need for additional bridging studies can be quite significant, especially there's a well-known example of a company that who were presented a dossier for a recombinant product to the FDA, and the master cell bank that had been used had been developed initially in a research lab, but they hadn't kept good records of the source of the raw materials that were used in the master cell bank, and because they couldn't confirm that the raw materials had been sourced appropriately, the dossier was rejected. So the FDA rejected the product. So they had to go right back to the start and prepare their master cell bank and then do extensive bridging studies. So setting up those systems early on is really important in terms of downstream consequences. Okay, so for phase 3, your manufacturing process should be well established and well controlled, and you should, as far as possible, use your formulation and presentation that you plan to take to market. And you should have sufficient stability data that you can manufacture the batch for the clinical trial such that each patient, each participant in the study can receive the same batch for the duration of their participation in the study and not have to change batches during their treatment period. So that, again, you're just reducing one source of variability during the study. And of course, by this stage in development, your documentation systems and your risk assessments and so on should be really well established. They should be established fairly early on, but by this stage, they really should be developed at the point where you're comfortable in presenting them as a product and a manufacturing program appropriate to take to market. Okay. So I just want to talk now a little bit about the research environment from a regulatory and industry perspective. And Duncan's already talked a fair bit about the industry, so I won't spend too much time on that, but just talk a little bit about the regulatory and ethics environment. So you're probably aware that we have two regulatory schemes under which clinical trials can be conducted. and as well as the ethics approval process. So under the therapeutic goods regulations, there's 4 mechanisms which allow individuals access to so-called unapproved medicines, so medicines which haven't been approved for marketing. So we're talking today about use in clinical trials, the CTN and the CTA scheme. The other 3 mechanisms, personal importation, SAS and authorised prescriber, we're not going to talk about today, just focus on clinical trials. Okay. So the regulatory framework for trials in Australia is quite different from all other competent jurisdictions. And the key feature is the Clinical Trials Notification Scheme, in which TGA has devolved responsibility for approval of both the scientific and the ethical aspects of the trial to the Ethics Committee. So the process is that the investigator submits an application to the Hospital Ethics Committee. So the Ethics Committee reviews the scientific design of the trial, assesses the risk-benefit, reviews the information and consent and the ethical acceptability of the study. And then together with the approving authority, which is the governance process, they will give approval for the trial. The sponsor then notifies the TGA through the CTN online portal, pays a small fee, there's acknowledgement, and the trial can commence. So from a regulatory perspective, it is very straightforward. TGA doesn't evaluate any data relating to the trial, although they may request copies of the protocol and the IB. I've seen that on some occasions. And they now have a GCP inspection program and may audit the study to ensure it's conducted in accordance with the principles of good clinical practice. But of course, that's once it's commenced, that's not pre-commencement. So at least 95% of trials in Australia are conducted under the CTN. And I say at least because we don't actually have any recent figures to confirm. TGA used to publish figures on it, but I haven't been able to access any recently. Okay. So the Human Research Ethics Committees in Australia are established under NHMRC guidelines. NHMRC has an accreditation scheme for ethics committees, including membership. But as you can see, only 2 members of the ethics committee are actually required to have research expertise. The other members may have research expertise, so your healthcare professional and your independent lay people. So, for example, I know one of the lay people on one of the ethics committees of our major public hospitals was previously a hospital pharmacist involved in clinical trials. So she has a good understanding, but she is there now as a lay representative. But there is no requirement that the other members would have that research expertise. So the documentation that's submitted to the ethics committees includes the protocol, the IB, the consent forms, and then the various administrative documents. But and if you think about the CMC, the manufacturing data that is included in the submission to the ethics committee, it's only what's included in the IB, and if the IB will generally include only maybe 2-3 pages of CMC quality information. So it might include some information on the formulation, the presentation, how to prepare the doses, very limited stability information, and a GMP compliance statement and names of manufacturers, and that's really the limit of it and the extent of it. So if a trial is conducted under the CTN, the CMC data is not independently reviewed. The Ethics Committee can request an independent review or seek additional data, but in my experience, this is really, if ever done. And I was speaking last week with a member of an Ethics Committee, and I asked him if their Ethics Committee ever requested additional data, and he said, well, they generally want to see the elements that I've confirmed are generally in the IB, including things like GMP compliance statements, stability information, and so on. But beyond that, they don't request or review any data. The Ethics Committee can also request review under the CTA scheme, but again, that's rarely, if ever, done. So the CTA scheme is the clinical trial approval scheme. It was previously known as the clinical trial exemption scheme, and you'll still see that in some documents, but the name was changed, I think it was probably last year, to the CTA scheme. So under the CTA scheme, the sponsor submits summary information to the TGA, pays a slightly larger fee. TGA will review the dossier, ask questions, etc. If they don't object, then that information goes to the Ethics Committee, and the Ethics Committee who is still responsible for review of the protocol and the ethical aspects of the study. Once they receive TGA's no objection letter, and provided they're comfortable with all the other information that's been submitted, will approve the study and the trial can commence. So those two processes can happen in parallel, but ethics generally won't approve until they have TGA comments. Okay. But having said that, the CTA scheme is mandatory only for Class 4 biologicals, and only if in certain circumstances. So, for example, if the biological has been used in previous clinical trials, or if that same trial has been approved by another comparable regulatory authority, so if the sponsor has an open IND, or it's been approved in Europe, then there's no requirement for the CTA submission. So you can see that the number of clinical trials in Australia where a submission is made under the CTA is in fact is very low. I made the comment that TGA evaluates summary information. They previously had on the website some guidelines under the old CTX scheme. When they changed to CTA, those guidelines were taken down, and so there's no guidelines available as to what summary information is required. TGA does, however, recommend pre-submission meetings, and presumably at that meeting, the requirements in terms of the dossier scope and content will be discussed and agreed upon. And I guess the rationale for that is because biologics and Class 4s in particular are generally novel products, and so it's difficult to write general guidelines, and so it's discussed and agreed on an individual product by product basis. Okay. So I'm not going to spend a lot of time on this slide because Duncan's already gone through it, but I just wanted to highlight a couple of aspects of the Australian environment that I think make Australia an attractive environment for conduct of clinical trials. It is accepted that we have because of the CTN scheme, it's a rapid startup, but sometimes we see delays in the ethics and, importantly, the governance approval process. So the advantages of the CTN, I won't say can be outweighed, but are tempered by the timelines for ethics approval. And I think we see this particularly in the public sector. In the private sector, the approval timelines are less. And I think if US companies are thinking about timelines for approval for studies in the US from an ethical perspective, they're thinking of the private IRBs, which have a very rapid turnaround, as distinct from some of the academic institutions and research facilities where the timelines are similarly long as they are in Australia. I think we do have a big advantage in having standard contracts, and American companies are always pleasantly surprised when I say to them we don't have to actually negotiate the terms and conditions of the contracts with the investigators, that we have that all that work's been done, and we have a standard contract that all stakeholders have agreed to use. We do have world-class investigators who are experienced in doing clinical trials, and I think really interested in clinical research, and institutions such as Peter Mac here in Melbourne actually aims to have 50% of its patient population enrolled in clinical trials. I don't know whether they've been able to achieve that, but it certainly has been one of their stated aims. So they really want to participate in clinical research and bring novel new products for their patient population. I think despite the significant challenges of dealing with the COVID pandemic, we do have a world-class healthcare system. COVID has created some challenges in terms of starting up new trials, and during the worst of the lockdowns for monitors even to go into hospitals to actually monitor studies and so on. Most of that has settled down now. I think the challenge now is the staffing resources in terms of setting up new studies, and hopefully some of that will be mitigated in the next few months as we somehow learn to live with COVID and get on with what we need to do. But combined with the advantages in terms of the regulatory framework and our investigators and our healthcare system, the costs are significantly lower than in the US and the UK, and for eligible companies, the R&D tax rebate makes it quite attractive to do studies here. This is just a representation of the information that was in the MTP Connect Trials report, so I won't go over it again, but I just wanted to emphasise and illustrate that, as Duncan said, that the highest percentage of trials in Australia that we're involved with are phase 1 and phase 2 studies. And I think that is because we do well at complex studies with requirements that require those world-class healthcare facilities, but our relatively small patient populations enable us to enrol patients for phase 1 and phase 2 studies that we generally can't meet the same recruitment targets as countries with larger populations required for phase 3 programs. So we do have some limitations. I think there's no doubt that patient recruitment can be slower in some indications and in later phase studies. I think there are some limited some challenges to our infrastructure in terms of our supporting resources, particularly for large-scale manufacturing, and we see that as an increasing challenge as Big Pharma has reduced manufacturing in Australia to a level that it's not just the products being manufactured here, but the expertise that is developed as part of scientists and staff working in those manufacturing facilities don't have the same opportunities to develop skills and expertise in the absence of that industry. And I think Australian companies do like to interact with FDA and EMA, and particularly FDA, because they have broad range of experience in assisting startup companies and are willing and able to provide advice to Australian startups, even if they don't plan to open an IND in the near future. It is possible to go and talk with FDA and get some and seek advice from them in a way, and because they have experience that because of our CTN scheme largely, TGA doesn't have that necessarily have that expertise available in-house. I just wanted to finish with a quote that caught my eye actually in yesterday's newspaper, and it was from a US actor to an Australian reporter, and as I said, presented in The Age yesterday. And I thought, well, that really is applicable to our industry as well. And the actor said to the reporter, I think everyone in the industry would tell you that your lot, that's Australians, are the best people to have on set. I think that is true across our industry, in that US companies, Europe in particular, and to some extent European companies, are really interested in working with Australian research facilities. And I think there is a real opportunity for us to continue to grow the industry, and as Darwin has said in his presentation. So thank you, and happy to answer any questions.
Fantastic. Sorry. So the question was, you mentioned quite early on the importance of including in trial design endpoints related to quality of life and health economics. Can you give us some practical examples of what that looks like, please?
So there are standard questionnaires for quality of life, and most, in fact, even in phase 2 studies now, we're expected to include those quality of life questionnaires. There are some general ones, which, but there are also quite disease-specific ones. So, for example, I'm involved in a food allergy study at the moment, and we have, and includes both adolescents and paediatric cohorts. So we have questionnaires that we get them to complete before they start treatment, and at the end of treatment, at the end of a follow-up period. And we ask questions about the impact of their food allergy on their life. And, you know, for teenagers and for kids, you know, if you've got a food allergy and you go to a birthday party and you can't eat the food, that's pretty tough on a young kid. So we ask them questions about the impact of the allergy on their social life, and, you know, how careful they have to be, and, you know, and at the end of the study, whether participating in the study impacted on their so, for example, you know, some of the food allergy studies, they need to be taking, you know, daily doses of medication. How did they accommodate that within their daily routine, and so on? So all later phase clinical studies these days, if you want to get reimbursement, and indeed even, you know, the investors want to see how the product has impacted on the participants' lifestyle and their concerns about their disease state, in addition to the harder clinical endpoints. And then that data can be used for economic modelling. And especially, you know, in conditions where if exacerbations of the disease, so in food allergy, for example, if a patient has an anaphylactic reaction because they unintentionally eat the food and end up in hospital, if you can reduce the likelihood of them experiencing an anaphylactic reaction and therefore reduce hospitalisation rates, then that's clearly a saving to the healthcare system as well. So you can develop economic models based on that data.
Thank you. And I would just highlight to everyone in the audience, online and in the room, that through our READY funding, we fund ARCs to provide training on clinical trial design, so it satisfies requirements of regulatory bodies, but also payers. And that will be running again in early 2023. Dominic, I can see that you have your hand up. Unfortunately, we will need to go to the next speaker, but there will be time at the end for questions. If you want to pop your question in the chat, that'd be great. I will introduce our next speaker, David Edmonds. David is former principal of CMC Regulatory, which provided regulatory advice to therapeutic goods R&D companies and manufacturers in regards to the regulatory and quality assurance of GMP requirements. He was previously Manager Quality Assurance and Regulatory Affairs at Peptech Limited for 20 years and held QA, manufacturing and laboratory management positions at Lilly, A.H. Robbins and Sterling Pharmaceuticals during the previous 20 years. David has been a member of the RACI Pharmaceutical Science Group for New South Wales from 1987 to the present. David, over to you.
Looks like it. Okay, good. Thank you very much, Duncan, and thank you to the audience and the Zoom folk for coming along. I want to talk about documentation and what resources are available. The therapeutic goods industry is very self-contained. It's got its own culture and guidance, etc., within it. And so what I'm doing is not the most riveting presentation you're going to hear this afternoon, but hopefully there will be some good use to you. And it's a listing of the various authorities and the various agency guidances that are out there, not private organisations, but the official agencies. It's too much to take on board in just looking at it, but I'll be putting this presentation up on the RACI Farm Group website, together with all the links that you will see. So the agencies first, the ones that are likely to be the most relevant, the TGA, of course, FDA, European, British, particularly after Brexit, Japan, and there's also an international coalition of agencies. And there are a multiplicity of technical guidelines. And these emanate from the Common Technical Document. And this document is for what's needed for final product registration. It involves, of course, safety, efficacy and quality. There's the ICH, the International Conference on Harmonisation, and PICS guidelines, the Pharmaceutical Inspection Convention. And there's the source websites for those organisations. So, like Judy, it's probably best to work backwards and look at what's needed when you actually register a medicine. And I'm looking, the link there is for the quality. And this is the context for an active material. And just highlighting the major paragraphs, general information, and name, etc. Manufacturer of it, how it's characterised, how the manufacture is controlled, and very importantly, the reference standard. They want reference standard details. More on that in a minute. And stability data. And the sub-paragraphs then go in. And the submission for this sort of one-page contents might go into numerous volumes. Likewise, the dose form, similar general requirements, the product, the formulation, of course, how it's made, and how the excipients are controlled as well. The reference standards again, containers and packaging, stability. Now, I might point out at this point, packaging is the sort of Cinderella of pharmaceutical development. Most important to protect the product and ensure that it's got as stable as life as possible. The ICH guidelines. Now, this is, again, safety, quality and efficacy. I'll be looking at the quality guidelines. This is an important link because the guidelines are in fact all over the shop in terms of the sequencing of them. So there's the general overview, starting with stability, which before analytical validation. So there's no, in a sense, there's no logic to it, but it's just the way that they're developed. Right through to 14 different major classifications. Then the detail and the more likely sequencing that you're going to need are the manufacturing system flows, and for following on, there's also a reference to the GMP for active ingredients, but you're more likely to be using the PGA guideline for that. The analytical parameters, which is the bulk of the guidelines. So again, procedure development. Impurities. Now, impurities are a major challenge in terms of pharmaceuticals. And the, excuse me, in essence, they can be classified into impurities related to the drug itself, solvents, and things like heavy metals that have been used as catalysts and the like. And there are guidelines for all of these, and in the early development stage, it's worth looking at these because these are impurities that have best to be eliminated later on in the scheme of drug development. Pharmacopoeias, you're probably more likely to go directly to the pharmacopoeia rather than to the these particular guidelines, but I've listed them anyway. So some of them are very specific test methodologies. So specifications, very important that specifications are established early on, and of course have to be finalised for drug registration. Stability testing, again, numerous sub-guidelines within the stability parameters. And a special area for biotech products. Okay, the pharmacopoeias, ones you're most likely to use are BP, EP, and USP, as Catherine pointed out. Pharmacopoeias are fabulous sources of information, not only for requirements, but just as a scientific documents, and especially the United States Pharmacopoeia. Pretty well anything that you want to know about scientific practice or the chemical scientific practice, you'll find somewhere in the USP. But they're expensive. The PICS convention scheme is where the codes of GMP are to be found. And as has been pointed out already, the Australian version is the version PE 00915. Nevertheless, for the record and for the sake of this talk, I've listed the website directly to the PICS. Okay, the TGA, Catherine has described in more detail the TGA activities. So a little of this will coincide with them. But again, I've got some of the references too that she's also mentioned. Legislation and requirements in 3 parts: the Act, the regs, and the therapeutic goods orders, which do vary from time to time. And it's worth, particularly in commercial products, it must be kept an eye on. clinical trials handbook, as Catherine mentioned. The Australian Register of Therapeutic Goods. This is all the therapeutic goods that are registered in Australia, can be found at that website. Terminologies, international scientific guidelines adopted. So there they are there. Now this is an interesting web link where it just talks about public PGA information. And within that has got the TGA licensed Australian manufacturers. So you can go to this website and it's there. You can search on the Australian Register of Therapeutic Goods and also the ingredients names and proprietary ingredients. And there's a shot of the Australian manufacturers website if you're interested, I guess. But that's where you'll find, if you're looking for manufacturers in Australia and testing organisations too, of course, then that's the site to go to. But we talked about the PGA GMP requirements, and TGA is currently using version 15. Version 16 of the PICS has yet to be introduced into Australia. And via the TGA website, you can get the introduction, the active ingredients, and noting, as has already been said, there's a chapter there on active ingredients for clinical trials, the dose form, and the annexes. And again, investigational medicinal products are listed in Annex 13, as Catherine has fleshed out. There's the TGA interpretation, and it's titled, as you can see. Some of its descriptions still refer to PE 00914, the previous version of the requirement. But if you go onto the actual links, they'll take you through to the PE 0015. There's a guideline there for it, and I understand it's being reviewed at the moment to totally take on board the version 15. But version 15 is the official version. Sorry, going the wrong way. So a few observations, if I might. In the manufacture of the active, you get, in essence, you get one shot at a clinical trial, and the failure is likely to be blamed on that it's a dud active. Where this might or might not be true. If the dose presentation isn't optimised, you might be less likely to have a positive outcome for the clinical trial. So you've got to work on the dosage and not just believe that the job's over when the API has been established. I see nods in the audience. The registration doesn't only depend on whether it's efficacious and it's safe. It's got to be able to be manufactured and purified and upscaled and to be stable in its own right under the right storage conditions and shelf life for it to be a fair income drug. Otherwise, it's a biologically active material. The physical parameters are most important. Particle sizes have got to be known, crystallography, etc., and they've got to be consistent from batch to batch. Jürgen will go more into this subsequently. You've got established synthesis from beginning to end to give the same impurities. You've done all your tox studies before the clinical trials are commenced. So if you go and change all your synthesis, you've got new potential impurities, etc., and it can be a potential problem. But impurities not only affect the safety of efficacy, they can affect the stability. And in this context, water can be considered a most unwelcome impurity in many respects. And no doubt other materials as well. And I've mentioned about the crystallography already. The analysis of these, of both actives and dose form, the data's got to be accurate and it's got to be numeric wherever possible. Your whole campaign to bring the drug to fruition depends on the quality of your data. So, and your justifications and conclusions are based on the data. So this means your data's got to be right. And the sooner you get your analytical and test methodologies right and established, and you can genuinely rely on them, then you're making real progress. Sorry. You also should be looking at manufacturing reference standards as early as possible, because instrumental analytical methodologies, and particularly quantitative methodologies, rely on the quality of your reference standard. So your analysis is arguably only as good as what your reference standard is, or the results are at any rate. And in that context, I mean that the reference standard is well characterised and well understood and quantitated. So we're going. It's important to have validated, as I said, to have this data put down the track, and for any patent applications, clinical trial documentations, project sales, collaborations, and hopefully your eventual product. So again, taking Judy's point, the sooner these things are established, the better it is for everybody who's got to work with the data subsequently, because almost certainly other people will be working with this data further down the track from what it was when it was established at the initial R&D phases. And so the translation of this data is terribly important. Documentation, I said that first paragraph in another way, and it should really be signed off, reviewed and signed off at regular intervals. So again, for patent purposes and for the reliability and for collaborations, etc., you've got it there and people can see that it's been professionally developed. As early as possible, make sure that the drug that you're working at, that you believe you've got, is actually the fair dinkum drug. I can't give you any specific examples, but I'm speculating, and I've got grounds to speculate, that an impurity of with a high activity can override what you think you might be working with. So make sure that your drug is known and characterised as early as possible. Again, we come back to the thing about the GMP requirements for phase 0, phase 1. I would strongly recommend to look at using GMP licensed people right from the very start of clinical trials. And especially if you're working with sterile materials, because this gives extra documentation, protection in liability claims, and probably enhances project collaborations and sales, and for use for approving further overseas clinical trials. So finally, the researchers, there are experts in all these different areas out there as consultants, and they're skilled in this area, and they can hopefully get you on the right track faster, but most importantly, save having to repeat work because it hasn't been done properly in the first place. And if you get right this sort of advice, it's eventually going to save money and save time. And this is what getting to the clinic, getting a product registered is all about. Good. Thank you.
Thanks very much for that, David. Do you have any questions for David? Let me stop sharing. We do have a question. Let me run over. Hang on.
Thanks, David. Less relevant for Australian researchers, but have you got any comment on the WHO guidelines?
No, I'm not. Oh, sorry. No, so sorry, I'm not familiar with them at all.
Do you have anything to add about method validation, of course? Because with, say, a phase 1 trial, I mean, you can really take validation as far as you like, and implementing something which is going to be put in place across a number of labs for a number of years is quite a different thing to doing something which is quite self-contained as a phase 1 clinical trial. So obviously, we're motivated to do validation is obviously necessary, but it's a matter also about how much people want to invest at that stage.
Yeah, and this is really the dilemma, isn't it? About the costs associated with these sorts of activities in comparison with the benefit. The guidelines as to how to validate are out there in terms of inter-laboratory validations. I think that can only be as circumstances dictate. I think it's got to be validated so that the data that's produced is reliable for future use and is also defendable. Now, if you do it with the formal USP guidelines or the ICH guidelines as to how to validate, then you've got a platform to be able to defend that data with.
Thanks very much for David. And the slides will be shared after this.
Yes. I'll be posting the slides up, but I think my email address has been circulated fairly well. So if anybody wants a copy of this presentation before we post, then I'm quite happy to send it out.
Thanks so much. Our next speaker is Professor Paul Young, CEO of Ab initio Pharma. Ab initio Pharma are a pharmaceutical research and development company specialising in the formulation and good manufacturing practice GMP production of pharmaceutical products in Australia. He's also Professor of Respiratory Technology at the Woolcock Medical Research Institute and has over 25 years experience in the area of respiratory product characterisation, formulation, development and commercialisation. Additionally, Paul is Professor of Commercialisation at Macquarie Business School, where he specialises in commercialisation, entrepreneurship, pharmaceutical, medtech and deep tech business creation. Paul has established, is on the board of directors of numerous small medium enterprises, including Nanopharm UK, where he was founder and director from 2000 to 2007, Oz UK Limited, where he's director, and Pivot Innovations, where he is also director since 2001. Over to you, Paul.
Yes, sorry.
We'll get there. Said I was a professor of commercialization, not of IT, so it's okay. Okay, obviously thanks to Pharmacy for hosting us this afternoon and for the teas and coffees and the events after. I'm sorry for the people who are listening in. You won't be able to join us for drinks after, but Ab Initio is sponsoring some drinks and food at the end of the day. So we welcome you all to join us next door. So I was asked to talk a little bit about taking molecules from the bench all the way through to the clinic and some of the issues that we have in doing that, particularly related to phase 1 trials and some of my experience. So what I've put together is a couple of slides or a few slides based around the strategies of a small medium enterprise or even an academic who's got a new molecule or a biomolecule who wants to take that through to the clinic, the challenges that are faced, the team that you need to surround yourself in order to take that drug through, as well as a case study of something that we've done through Ab Initio. And I believe Jürgen is talking next about the active pharmaceutical ingredient. I'll be looking more at the product. But as David said in the previous talk, getting the actual molecule at the beginning, the API, in a high purity form in a GMP environment, I think is also a critical thing in the whole process. The number of clinical trials that we've been involved in where we've started doing development work with the API and the purity is not really well known, and that you end up with trials failing at an early stage if you also don't include the concept of using GMP at an early stage for your API as well as your active pharmaceutical ingredient, as well as your product. So who are we? So Ab Initio is a pharmaceutical formulation R&D company, which was spun off from the University of Sydney. It's in partnership with Royal Prince Alfred Hospital, Sydney Local Health District, as well as MTP Connect and ARCS. And our mission is actually to help small, medium enterprises, academics and clinicians take new molecules or biomolecules, turn them into workable products that they can then take through to clinical trial and then help them scale that through to later clinical trials and commercialization. We're an independent company, which means we're agile. We can work with small and medium enterprises throughout Australia. We also work with companies overseas in the US, China and Europe. We were established in 2019. We've got around 18 to 20 staff, including staff at our GMP manufacturing facility and pretty much 50-50 GMP and R&D. So what we actually do as a company, we help people take a new molecule into a clinical trial. So what we tend to find is an academic or a molecule which would be licensed out of a university to a startup company. They have an indication, but they don't really have a dosage form in which they can take that indication and turn it into something that's actually useful. So for example, a therapy for neurodegenerative disease where they've done testing in rats, for example, where they've been injecting the drug into rats' brains, that's not going to be a suitable product for a clinical trial. So we can help them undertake the initial R&D, research into actually turning that into a dosage form where we can actually, whether it's a tablet or a nasal drug formulation, where it can actually be taken by a patient. The other thing we actually then do is look at feasibility of developing that and the concept of how we're going to scale that to a manufacturability process, which we can then move into GMP for a clinical trial. The other thing that's quite important here is in terms of this whole process development, and this is not about Ab Initio, this is just in general for people who are formulating new molecules. You obviously have the medicinal chemistry or the biochemistry part of drug development. But the other thing you always have to consider is the actual formulation itself, as David has already and Judy has sort of said. The formulation plays quite a big role, not just in the delivery vehicle itself, but also in the intellectual property. If you look at intellectual property around commercial therapeutics, a lot of the IP is based around the dosage form, not just the active pharmaceutical ingredient. So you've done all the work as a medicinal chemist or you're working on a biomolecule. and you've discovered a new drug or a new therapeutic, you have an indication for that drug, and you want to conduct a clinical trial. So what do you do next? And this is just some key thoughts that I have around this. So I think the most important thing is you have to understand that, you know, your drug is not the end product, and you have to turn that into a workable product. So whether it's an RNA molecule for a vaccine, you know, as we know, the development of the Pfizer vaccine required a very complex formulation, and it's got a -70 cold chain process, right? At the same time, if you created a small molecule like, this is just psilocybin, but you had to decide on a dosage form for that. So does a patient take an oral formulation, or would, you know, nose to brain be a preferred route? So you have to think about the formulation that you're going to use this drug in, and the challenges that are going to be faced. And ultimately, those formulations themselves, as in the Pfizer technology, will have intellectual property associated with it. I can't say this more than enough, to be honest with you. Again, this is what David said. I think you need to start thinking about the actual formulation from day one and the product from day one. The companies that we work with, and they tend to be small, medium enterprises and startups, they all work towards a pre-IND meeting and investigational new drug product, or an IND submission package, and they start from day one. Don't think that you can make a drug in a chemistry lab in a pharmacy school and then turn it into a clinical trial. It's not going to become a product. You're going to fail very, very early on in terms of your quality management system, the documentation, the API, the assays that you're using. Just from day one, once you've got some IP in terms of your molecule, start looking about how I'm going to manufacture this to GMP standard as an API. What documentation do I need around that molecule? So do I understand its stability? Do I understand its degradation products? What's its related substances? Does it interact with the environment, and so on and so forth? And having all that key information in the chemistry manufacturing controls package that will go into the IND is essential. Do that at the beginning, because otherwise you'll just be backtracking. Don't try to do the clinical trial and then backtrack and do everything else, because all you're going to end up with is generally just fail your clinical trial. Identifying a dosage form at an early stage is really important as well. So probably rather than just go into a phase 1 trial without your final dosage form, try to get as close to your final dosage form as you can. So don't do a new drug molecule for Alzheimer's with an oral tablet when your actual your final product is going to be a intranasal drug delivery vehicle. Much easier just to start with the formulation that you actually intend to be using at the end. Try to identify formulation development expertise. There is plenty around, and CMC expertise. So people who understand exactly what David just said, understanding the chemistry of your molecule and the degradation processes, the related substances, and so on and so forth. Always consider PICS-2 and PICS-1 in early stages. So that's the API manufacturer and the finished product manufacturer. And have a clinical trial strategy right from the beginning. And the reason why I say this is already important is because of the sheer costs. So Australia is a good place to come to do a phase 1 clinical trial. And it's not because we have exemptions. As I'll say as we go through this, I believe there shouldn't really be exemptions for phase 1 trials for GMP manufacture. But what is good is that we have great clinical trials infrastructure in Australia. We also have tax 43 cents in the dollar for Australian companies in tax rebates to undertake clinical trials. And clinical trials cost a lot of money. So this is from a US Department of Health review. And as you can see, for phase 1 trials, this is the sort of US dollar amounts you're looking at as a phase 1 trial in the US. And it's not too dissimilar, but it'd be Australian dollars here for each of these different categories. So as you can see, to do a respiratory drug phase 1 trial properly, it's probably going to cost you 3 to 5 million. You also need to consider at the same time that you have to do the preclinical properly. And that's something that we don't do in this country very well either. So getting the GMP API material for your animal studies and getting that done in a GLP environment is absolutely key, because having it done in a laboratory in the basement of a university somewhere is not really going to fly when you come to submit documents to the FDA. They're just not going to look at it. The documentation won't be there. The number of animal studies won't be there. The methods won't be validated. You have to think about the preclinical study from effectively a GLP point of view. As I said, preclinical in vivo costs of generally 3 million. So that's what your animal cost would look like. Your clinical trial cost is probably going to be equal, if not more. As I said, the costs of doing these trials are extremely expensive. And the problem and the reason why trials fail is not necessarily because of the molecule itself, but more than often, just poor manufacture of the API, poor choice of dosage form, and poor manufacture of the product for the trial. So I was involved in a trial way back when I was in the UK, and it was looking at a phospholipid for delivery to the lung. And it had a glass transition temperature of 37. And the entire trial fell because of poor manufacturing, because it was not done in a GMP environment, and it cost them millions and millions of dollars. So considering all these things up front is really important, because otherwise you'd just be wasting money. And obviously, a lot of people, seeking money either from government bodies or from venture capital. And the venture capital people are not going to be particularly happy if your clinical trials fail based on the fact that you just had it made in the basement or down the road at Chemist Warehouse. It's important to consider also the market and registration. So your pre-IND, IND meetings, and the documentation that surrounds those. The same would go for the TGA and the EMA. A lot of people that I work with in Australia generally actually target FDA initially. And that's more to do not with the fact of ease, but just to do with market access and health economics. So some essential skills that you need to surround yourself with. So the first one I put up there, that's actually quality management systems. And it's really not QMS systems per se, but just the importance of documentation. So the audience here is quite wide today. We've got companies, but we've also got academics as well as SMEs. And understanding good documentation, even before you start going down the road of GMP, is really, really important. Academics particularly are not the best at documenting things properly. And that really impacts your clinical trial. But more importantly, it also impacts your intellectual property. And having done expert witness cases over the years, IP that comes out of universities is horrendously problematic because the documentation in the initial intellectual properties not done correctly. So understanding your documentation is pretty key. Having people who have strong understanding of intellectual property is important. People with regulatory experience, understanding how to develop a TPP, preclinical expertise in tox and PK, and actually undertaking proper high quality preclinical tox data before you go into the clinic. Preclinical tox data is not just important for documentation, it's also important for the sponsor's liability. So if your animal work's not done properly and you have an issue in your clinical trial, and it may come back to the sponsor because the animal study wasn't done properly. And the same goes with the manufacture of your product and the manufacture of the API. If something goes wrong in the clinical trial, the liability is ultimately on the sponsor. And if the sponsor has gone to a compounding pharmacy and manufacture granules or tablets for clinical trial and has not done any quality control on the release rate or the dose uniformity, or the purity or impurities of the API, and there is adverse side effects, that liability risk is absolutely huge. Expertise in chemistry, manufacturing controls, industrial chemistry is really important from an API point of view. I guess as Jürgen's going to talk about in a while, scale up is also something that you have to think about from day one. The formulation, obviously, as I've talked about before, understanding GMP, understanding scale up, and then these parts here that people don't really think about. If you're an SME or a startup or an academic, understanding business, organizational structure, business management is really important. Understanding the financial and market modeling for your product is really important. If it's no good coming up with a medical device, which is going to cost $600 per unit when your competitor's 6 cents. The same would go with that for therapeutics. As well as corporate finance, understanding how companies work, how shareholders own parts of businesses, how venture capital works, and so on. So where do you get money from? Well, funding comes from multiple avenues. Depends on who's listening to this today. So obviously we've got academics, small, medium enterprises and startups. There are lots of opportunities for money around, including MTP Connect. There are incubation and accelerating programs all across Australia. There's venture capital funding, license and partnership, which universities obviously attempt to do with intellectual property that's developed. So the IP gets licensed to a larger company that then takes that through. General government funding, such as minimum viable product grants, and then of course things like NHMRC and MRF funding. NHMRC development grants, for example, are pretty good at taking drug molecules through to clinical trials. And where my historical academic hat, I've conducted a number of development grants over the years, taking products through into phase 1. So just a couple of things around phase 0 and 1 exemptions. So obviously there are exemptions, although looking at it from a liability point of view, and also looking at it from even from the TGA's clinical trials handbook, there are a couple of things. First one, initial experiment studies in human volunteers, really focused on pharmacology. So the idea of, say, taking a repurposed drug and then conducting a phase 1 study, is that exempt or not exempt? I mean, this is a question we get asked all the time. I would probably say not exempt, because the pharmacology has already been done, and it's already been done in a limited number of subjects. So the idea of repurposing drugs, I'm not necessarily convinced on. But either way, I think generally the important thing is this: the trial sponsor should ensure that they have thorough understanding of the principles of GMP in order to assess the quality of the therapeutic goods. And so getting something compounded down the road and not doing any quality control or having any QMS system, and the same goes from the API, drives this significant liability for sponsors. Things that you need to consider, and I'm just going to go through these very quickly because everybody else has been talking about these, and Jürgen's going to talk about API. So characterization and understanding your API is extremely important. I'll share these slides on the RACI website as well, so people can look at these at their own in their own time. Quality control, so understanding your incoming materials, very, very important. Robust sampling plans and testing of the finished product is essential. And having validated analytical methods. So not just a UV or an HPLC method for looking at your total drug content. You also need to know all your degradation products or related substances, because as David said, a related substance in the drug may be more potent or more toxic than the drug itself. And then understanding where you're going to get these molecules made. And obviously the TGA licenses facilities for clinical trials material. So you have to look at a facility that has that license. And then you also have to consider what you're making. So for example, are you making Schedule 4 drugs? Are you making Schedule 8 or even 9? The psilocybin example that was a few slides back would come under Schedule 9. And then is the facility licensed to do all steps? Can they do blinding? Can they do the quality control on the of the actual product that's actually leaving the manufacturing site? And then lastly, you need to consider technology transfer. So again, speaking to numerous people over the years, something in a lab where you end up with being able to produce 1 microgram of 2 nanometer particles necessarily doesn't really scale to commercial manufacture. So when formulations are developed, having a strategy for, can I actually make this? You know, if I can make it in a lab, but can I make it at scale? I think having scale up and feasibility at an early stage is really, really important. Okay, so this is just a very brief case study of something that we've done recently. We deal in all sorts of dosage forms, from sterile injectables through to capsules and granules, but we also do a lot of nasal and inhalation products. And this is just an example of taking a new molecule through to clinical trial and the importance of quality systems, quality preclinical data, and a strategy for that clinical trial. So this was based on a small molecule. We started this 12 months ago. The clinical trials commencing now. Novel small molecule. So initial feasibility work's done in non-GMP material, but the first thing is to get GMP API, because what you find is, as David says, the initial chemistry lab material, strangely enough, is not the same as the GMP material coming from the API lab. And so you want to make sure you get high quality material. The drug had good liquid solubility, but very poor oral bioavailability. So we chose a respiratory, or they chose a respiratory pulmonary target. And it was actually designed to stay in the lung. And it's also designed as a subacute indication, not chronic. So the first thing is, if you're doing a clinical trial in phase 1, so that single or multiple dose escalation study, the first thing you've got the problem with is, when you're making a product for a phase 2 or phase 3 clinical trial, you actually have a product which is a fixed dosage form, right? So it will be a 100 milligram tablet, for example, or 10 milligram. In a phase 1 trial, you're doing a dose escalation. So what you've got to do is you've got to find some way that you can create a formulation which can go from very, very subtherapeutic target levels, because you're looking at toxicity, up to and maybe slightly higher than your targeted therapeutic window. And so that becomes very challenging, particularly for a respiratory product, because if I took an inhaler like this, which had a 100 microgram target dose, to do a clinical trial where I'm dosing people from, say, 0.1 microgram through to 100, I'd need to make dozens of different formulations, which is dozens of manufacturing runs for a clinical trial, because each batch that you make is effectively a separate product and a separate QC and release. And the same goes for placebo. That's the other thing that people need to bear in mind. The placebo is no different than the product. So when you make a placebo in a GMP environment, it's exactly the same as the product in terms of the quality control and the systems. And it's not just, oh yeah, we make the drug and now we make a placebo, and oh yeah, we can just do that on the side, that's no problem. It still has the same rigor. So the concept was, we'd develop an initial formulation as a nebulizer, and then build that in so we can do the preclinical GLP animal trial as well as the phase 1 trial. And rather than create dozens of different formulations with different doses, because 1 microgram dose, 10 microgram dose, up to, I think it was up to 6 milligram dose, 6,000 micrograms, would mean you'd need to make different products for each dose in the trial. And so what we've decided to do is, using a breath actuated nebulizer, we could create 2 formulations where the patient would be dosed on the number of breaths that they have in the trial. So this is a brief overview of the target. So a GLP study was conducted in small and non-human primates in a GLP environment. Again, this was done in the US, and it was being used for an IND submission. So all done with a great quality management system. The formulation was identical to that was going to be used in the humans. So of course the FDA are very happy with that. Although dosing very small animals is problematic. However, this is doable. And we needed a wide potential dose range for the target receptor. So we had a 100-fold dose range. And so basically what we did is create 2 formulations. And again, now this goes back to what you need to do. So in terms of the CMC documentation, understand the solubility of the drug, understand its related substances, establish analytical methods, understand physical chemical properties of the drug. So everything from its thermal properties, crystallinity, what else would we do? Moisture absorption, etc. developed a number of prototype formulations, established stability of the drug based on the related substances methods, right? So you need to know what the degradation products are. Does the drug interact with oxygen? Does the drug interact with light? All of these things have to be tested for, and then narrow that down to 2 lead formulations. So now we have 2 formulations that we can manufacture for the clinical trial. One is 0.1 mg per mil and the other is 3.5 mg per mil. And that would, under a breath by breath process in terms of drug delivery, that would give us a range that could take us all the way from literally tens of nanograms up to milligrams. This is the aerosol particle size distribution of those 2 formulations. So they are suitable for respiratory delivery because the particles are less than 10 microns. This is some initial trial concepts. So this isn't broadly following the United States Pharmacopoeia. What we were doing is looking at if we nebulized this using a breathing simulator, which we've got as a qualified piece of equipment in the laboratory. If we had 5 breaths or 10 breaths over different breathing periods, would the patient or the subject for the clinical trial receive the same amount of drug as a function of time? This is just some of the stability data. So, right, go back. This is just some of the stability data. So we looked at, this is just pH. We've done pH, but also related substances as a function of time, because we have to control the pH of these nebulized formulations as well. And this is done under different ICH conditions. And then we move on to United States Pharmacopoeia testing. So what we'll do is inhaled nebulizer studies using impactors, which are mounted in these fridges as per the USP recommendations. And this is actually V, one of our laboratory scientists doing the testing there. And this is the particle size distribution of the lower dose formulation. And this is the particle size dose range of the upper. So all this shows is the size categorization of where the drug will go into the lung in the clinical trial. And this is the summary table that's then included in the clinical trial documents. So GST, the fine particle fractions, the percentage of particles that will go into the lung. So we've got equivalent percentage lung deposition irrespective of the dose that we're delivering to the patient. So now we're in this position where we can dose patients from, say, 10 nanograms all the way up to milligrams, and the percentage of the particles, i.e. the bolus into the lung, will always stay the same. So now we've got equivalency. And we undertake then breath simulation studies. This is a breath simulator. And what that does is then cycle, again, under United States Pharmacopoeia outline of what you should test. So how many cycles per minute would be an adult. And what we can then calculate is the number of or the amount of drug that the patient is going to receive as a number of breaths in the clinical trial. And what you find is that there's a direct linear correlation between the number of inhalations that the patient receives and the delivered dose that they receive and the delivered lung dose that they will receive for both the 0.1 milligram per mil and the 3.5 milligram per mil. So now we can go into a clinic and we have 2 formulations rather than 20, as well as a placebo. So if you have to manufacture 3 formulations and release those for QC, you have 3 formulations, then go into the clinic, and the patients then in a SAD or MAD study would then just inhale the number of breaths that you require them to inhale to receive that dose. And that's just the consistency of dose per breath that the patient receives as a fixture of breaths. So it's basically linear, which shows that's a very, very stable formulation. So in summary, we undertook a preclinical or developed a preclinical data package. This is part of a larger presentation. So I had a colleague in the US from a company called Coleus and Lovelace who undertook the animal studies. So we undertook preclinical data package, formulation package, initial CMC work. And what we actually did then is fast track that to first in human study, which is actually, I believe, commenced already. And the formulation was developed specifically for dose ranging, but at the same time was also developed in such a way that it could be literally the ideal dose could then be plucked out of that effectively formulation and then put into the phase 2 trial. So effectively, this would then go on to be the final part of the final product. And that's just our team. Thank you.
Thanks very much, Paul. Do we have any questions for Paul? I have a few quick ones. Oh, hang on, Alan, I'm going to ask one quick one and then I'll come up to you while Paul answers it. Paul, you mentioned lots of different areas of expertise. Where can people start to access some of that kind of information?
Yes, well, there's lots of expertise. There's lots of expertise in consultancy companies around that can offer that support. There's also training programs as well, as you would know. I mean, MTP Connect. I mean, there's significant information there that people can glean in courses on everything from GMP to regulation. But generally, what we do, particularly with small medium enterprises and startups, they tend to work with consultancy firms that then will help them start to piece together for that product.
And can Ab Initio help out in that respect too?
Yes, we can. We don't have all expertise, but certainly the CMC part, the formulation design, helping with sort of the early sort of concepts of the clinical trial. I just gave, I mean, that was our concept to overcome the challenges with having to create so many formulations for a SAD study. But ultimately, you also need, PK experts. You need, clinical trials expertise as well, and people with access to, for example, pre-IND meeting. You really want to, get in contact with people who could do that. And we have really good contacts that can actually. We actually work quite a lot with specific companies that do that IND document prep. IND document prep.
Yeah, you made the comment in the talk that for the phase 1 clinical trial, you're going to try and get as close as you can to the final dosage form when you put that into trial. Just this is outside of my area of knowledge, I guess, but I would have thought if you have a new molecule, the first thing you'd be wanting to find out is whether that molecule works at all. And then you tend to just do injectable and then maybe in another trial, optimize the dosage. Because I've always seen more injectables come through the testing for phase 1 work. And I've always assumed it's because the first thing they're doing is seeing if the molecule works.
I guess it depends on what the indication is as well. So, I mean, if certainly respiratory, it makes no, I mean, yeah, I don't know. I think I personally believe it's worthwhile developing your dosage form for the. So like if you're looking at respiratory infection, you would want to deliver a respiratory antibiotic, right? You wouldn't want to deliver a systemic antibiotic and then hope that it basically goes through the systemic circulation and then back onto a topical surface. The same as if you had, a wound healing drug, you would have a dosage form for topical administration. And certainly with things like dementia, neurodegenerative diseases, trying to target those is always a problem. Particularly that's one of the issues is translation from animal to human, where a lot of the preclinical work, particularly where people are doing phase extensive preclinical small and larger animal models where they're then just saying, okay, well, it worked by injecting it into a rat brain, and then they haven't done formulation development program to show that actually does work if they deliver it like intranasally and then in an animal and then move to a human. So I would probably lean toward dosage form first of all. Yeah.
Thanks, Paul. Please join me, everyone, in thanking our 4 speakers again. We've had some fantastic presentations so far today. I guess one of the, sorry, please. I hope one of the messages that's come through really strongly is this idea of begin with the end in mind. And for anyone who's sort of on this journey, getting towards this journey, please do take that to heart. I think it's come through really strongly from all of our speakers. We're going to have a brief break now. It is currently 3:15. We're going to come back at 3:35. So there will be some afternoon tea next door for those of us who are here in person. And then we have 2 more talks after the break. So please join us after that. Thank you. All right, thank you very much to School of Pharmacy for the tea just then, very much appreciated. Our next speaker is Dr. Jürgen Lindner. Jürgen has extensive experience in the manufacture and regulation of therapeutic goods, from early development stages through to commercial manufacture with a focus on CMC, quality management and compliance. Over the last 30+ years, he's held several scientific and senior management positions in both academia and in the biopharmaceutical industry, and is a consultant to this industry. Jürgen graduated from the University of Heidelberg with a PhD in neurosciences, and he has been a committee member of the RACI Pharmaceutical Sciences Group in New South Wales for over 25 years, and is a founding member of the Active Pharmaceutical Ingredient Manufacturers Association of Australia, API MMA, founded in 1997. In his position as the Executive Secretary of API MMA, he has been involved in the Australian TGA review process for the drafting and implementation of the Code of GMP for APIs, ICHQ7, with annual GMP update meetings with the TGA, and has been a member of the TGA Industry Working Group on GMP since its implementation in 2014. Please join me in welcoming Jürgen.
Thanks, Duncan. Well, we've had plenty of presentations telling you about what you have to watch out for. If, as they say in, was it Alice in Wonderland? If you ask where you want to go, if you can't tell me where you want to go, I can tell you how to get there. So in this particular talk, because a lot of the actual GMP requirements, the licensing aspects and compliance aspects have already been addressed, I'm not going to go into the details of the GMP requirements that apply to manufacture of APIs, but rather focus on when you're looking at developing a new molecular entity, at what stage of development do you have to have what type of activities and knowledge in process. The guidelines, again, I'm not going to repeat again. David has given you in his presentation the full detail of the guidelines that are applicable. Yeah, and I said Craig is in the next presentation, he's going to deal with the requirements for the actual medicinal product, and Catherine has already addressed the general GMP requirements. So what do you actually have to consider when you're moving your new, I call it actually an ME, because it's rather either you talk NCE for chemicals or NBE for biological, so I just left it as generic as possible for this talk, covering basically the general principles that apply to any sort of new entity that you're trying to bring into clinical trials. What you want is a commercial product that you can register. So as a base, you might say what all these products have in common is you have requirements for quality, safety, and efficacy. If either of those parameters is not met, you haven't got a commercial product and you've wasted your money in the development process. Key risks that you're facing there is that you have very limited safety information in humans, sometimes none at all. The process that you're dealing with is usually at lab scale by the time it comes out of R&D, so it's very small scale. You have very limited knowledge about impurities in the structure of your new entity. Characterization is incomplete, and you've got to deal with patents and IP protection. So when it comes to patents and IP, it is vital that you protect your gained knowledge as much as possible. Any lawyer will tell you that the value of a patent is only as much as you're prepared to pay for its defense. So sometimes a patent is not the best way to keep information in your knowledge sphere. What you can do, and I don't think we have addressed that yet, there is a thing called a drug master file, which is what you have to submit with any sort of applications for registration. The drug master file contains two sections. There's an open section and there's a closed section. any sort of, the open section is available to anybody who is able to access it, your customers, for example. But the closed section is confidential between yourself and the regulator and its evaluators. So any sort of critical information about your process that is essential for achieving the right quality parameters, you would put into the closed section of the DMF. So that's one way of protecting your IP as well. The risks, I'll just go through the risks that you're facing at the early transition stage. First of all, it's already been mentioned, is reproducibility. You don't know whether or not at this late R&D stage, the manufacturing process can be fully scaled up. The way to address that you're not missing out on anything, because a lot of, as previous speakers have mentioned, a lot of cases, drugs have failed because there was inadequate information or the final efficacy products, efficacy profiles were not achieved with material that was then manufactured commercially. The best thing what you have to consider is that you make sure that all the processing conditions are detailed and documented. The materials that you have used on how the product has been synthesized, or if you look at biotech products, the cell banking that you've used, the materials that you use for the feeding, anything that is used in the process should be documented. The equipment you have used, the equipment running conditions, analytical methods and equipment should be fully documented. The manufacturing entity needs to be characterized, so you should get information about the structure, the activity, purities and impurities and related substances. And I think David has raised that already, you need to define what a batch is. At the early stages, before you actually get into the clinic, you have to conduct your GLP studies, tox studies in animals. If you use multiple lots of R&D scale produced material and you pool that, that is then defined to be a batch. Yeah, sometimes you may use a pool batch, sometimes you may use individual batches. It is important to document what you have actually put in, because that will set the pace for where you're going next. The process, ask yourself, can the process be scaled up to commercial? If you have a small scale process that only works in the laboratory, you haven't got a commercial product at the end of the day. Sometimes you may use certain processes and that work very well in the laboratory, and you think, eureka, I've got a process. But then you look at it and say, okay, if I scale this up, what's going to happen? I'm across a couple of cases where they used flammable solvents, which are okay to use in the lab scale. But when you think about scaling that up to a few thousand liter, all of a sudden you have to consider that your facility needs to be explosion proof, which if you're organizing your own facility adds a substantial amount of cost. And if you're looking for a contractor, it means the contractors will be limited. Are the materials that you're using in the manufacturing of the API suitable for pharmaceutical use? If you can, I suggest that you actually use pharmacopoeial grade reagents all the way through from the beginning, because if it's pharmacopoeial grade, it's going to be known to regulators and it's going to be registerable. Unacceptable solvents is another issue that can arise. The ICH Q3 guideline deals with the limits of solvents that you can use in a manufacturing process. Certain solvents, usually the ones with carcinogenic potential, are pretty much eliminated. I mean, if you can stay away from using any of those solvents, do so, because your path to registration is going to be so much easier. On the biological side, you have to consider contamination with bovine spongiform encephalitis, viruses. I mean, there's all the guidelines out there which tell you what you have to look for. Pesticides, mycotoxins, and herbicides usually apply to plant-based materials that you may be processing, and elemental impurities apply to either of those categories. If, for example, in the purification process, you use, let's say, certain column materials, ask yourself, are those materials available at a commercial scale? It's no use if you have a production process at lab scale that you can run, and the material or the particular column that you use to purify your substance is not available at a commercial scale. You haven't got a means of manufacturing it. The whole aim of this exercise is to make sure that you avoid any manufacturing process changes at the GMP stages. Because, as was mentioned before, the material that you use in your preclinical studies, which comes out of R&D, is the key driver for quality, safety, and efficacy. If anything gets changed, if any of the impurity profiles or related substances profiles changes, you may impact on the safety of your compound and either have to go back and do further studies or your R&D gets completely rejected. Documentation, the pet horse for anybody who loves GMP. Good documentation allows you to do a thorough investigation of any failures. If it's not documented, how do you know where you can find a failure afterwards if you find that the batch didn't work? It facilitates process improvements and also tech transfer. Consider when you're moving from the R&D stage into the early phase manufacture, the company that wants to manufacture your product will want to know, how do you do it? Do you have documented process conditions, or do you have to reinvent the wheel? Good documentation helps you to smooth that process and also think about liability. It documents that you have taken due care. Because, as the legal auditor will tell you, if it's not documented, it wasn't done. If ever anything goes wrong and you get pulled in front of the courts, if you haven't got the documentation there to prove that you did the right thing, you've got a bad chance of going through. Okay, last not least, the most important one, safety. The key risk is that you have with any sort of new molecular entity in a phase 1 study, you have absolutely no previous human safety knowledge. It's all nice and well to say, okay, I've done animal trials, but you know, it's what you can say is there is a Pigeonero case where they developed an antibody against CD28, which is an immunomodulatory agent, and they developed a drug in 2006 for B-cell lymphoma as well as arthritis. When they applied the drug, which was manufactured under GMP, which was evaluated by the MHRA, they applied it to the clinical trial volunteers, and 6 out of the 8 ended up with severe systemic organ failure and serious problems. The investigation showed that all the documentation that was provided by the regulator as well as by the manufacturing company was in order. They were not at fault. It was simply an unknown entity. Can you imagine, with these sort of patients being in hospital for months on end, what sort of liability you'd be facing if you can't prove that you have done everything according to the book and have taken due care in your process? So from that perspective, even though it is exempt from GMP compliance, consider, is it worth ignoring the GMP principle with the risk that you're taking? You know, it's just seriously an issue. Good manufacturing practice, yes. What's it all about? It's just another code, which a lot of people say, okay, well, I'm wasting my time, I've got too many regulations. But I remember one of the first lessons I learned from there was a Lloyd's auditor for ISO 9000 compliance, and he said, you know, it's just not a case of compliance for the sake of compliance. Look at what the code can do for you and how it can improve your business operations. And if you don't get a benefit at the bottom line, you're not applying the code correctly. In the same way, I would look at GMP. Yes, it generates more paper, unfortunately. But at the same time, the principles of GMP provide guidance for managing these risks that you're facing at this early stage, and they provide a good structured science approach for introducing and managing any changes. Key objective, as mentioned before, is quality, safety, and efficacy of the product that you're producing. So what actually are the parameters that ensure product safety and efficacy? First of all, it's your product knowledge that you gain in the development process from the R&D stage. Then you've got the materials. The materials that you use in your process bring in certain qualities of composition, which means either purity or impurities. Manufacturing process has an impact. How you make your drug will define whether or not you end up with a safe and efficacious product, and whether or not, say, if you take a chemical synthesis I'm not that familiar with, but say let's take a biological, Is your product produced in the right conformation? Does it have the right post-translational modifications, etc., etc.? So your process has an impact in that respect. The equipment that you use has an impact. If the equipment is not calibrated or not monitored, you end up with variable inputs that you can't control. Manufacturing environment. You see some lovely slides from DGA inspectors occasionally. There was a beautiful one from the old, old inspector of Tribe. He presented a inspection of an API plant, I think on the subcontinent somewhere, where the photo showed pigeons roosting above open containers where the API was manufactured. Guess what was deemed to be inappropriate environment? The other thing, even if you're looking at a good facility, if it is a multi-purpose facility, you have to consider that potentially the dust being generated in the production or drying process can go through the HVAC system and contaminate your product if you haven't got the appropriate controls in place. So the environment is important, and last not least, it's people. If your people change and they don't follow procedures, it will have an impact on the safety of your product. Essentially, when you look at the red arrows, all those chain aspects, if you make any changes there, they have the potential for impacting on the product safety and efficacy, either by introducing new or elevated impurities or ending up with a faulty product. So these are the points that GMP essentially decides to control. So in summary, basically, the risks that you need to be managing at that stage are process and specification changes. Early in development, you haven't got the full process established, so you're still working your way around, getting a feeling how the product will behave, how the processing parameters will behave, so you end up with trying to improve your process, and you make changes to the process as well as to the specifications as you go. Quality activities need to be defined and documented, and most importantly, also for your IP, the knowledge that you gain in the development phase needs to be extensively documented. Specifications have already been discussed. The products that you use in the trials need to meet predefined requirements for quality, purity, and safety at each trial phase. They're not given from phase one. You go through, and I'll go through it at a later stage. They change throughout the development process. The level of GMP control also increases. It's not expected that at phase one or phase two, you have the same level of GMP control as you would have in a phase three or in commercial. If you put controls into your process, whether or not you implement them, you should document and justify them. I mean, there are cases out there where somebody early on says, okay, well, I've got this new compound. I don't know how stable it is, so let's put it at -20 in the fridge, in the freezer, and it'll be stable for good. Nobody takes any action. It goes through the development process, and all of a sudden at the registration stage, it states product must be stored at -20 degrees. Whereas the work that you've done in the meantime shows out that the product is quite stable and happy at 25 or 30 or 40 degrees. So all of a sudden, if you don't take care of those items and review your knowledge as you go through the phases, you may end up with conditions that make the product less commercially attractive or viable. If you have any deviations or out of specification results, don't be afraid. They are expected in the early development phase because you make constant changes. The important thing there is to make sure that you investigate these deviations thoroughly and gain more product and process knowledge. GMP starts with the introduction of the starting material. I don't know if you can read that. This is just a copy from the, what is it, page, I don't know, early in the part 2 of the Fixed Code of GMP. But what you can see, the gray highlighted areas are the ones where, the GMP starts. For example, for a chemical, it starts with the introduction of the API starting material into the process. The API starting material for a chemical is defined as the key chemical compound that forms the major backbone of your API. For a biological, for example, it starts, as you can see down here, with the introduction of the material via the working cell bank. Throughout the development to the final purification of the finished API, it is expected that the GMP requirements increase and you have tighter control over the materials that you're using. Some companies initially, especially on the chemical side, may have thought that they're being smart by outsourcing the production of the API starting material and then introducing something which is about 1 or 2 steps, or maybe just 1 step away from the final stage. FDA quickly caught onto that and said, sorry guys, there is something like the propinquity rule that we apply, and the definition of the starting material is that it has to be at least 3 synthetic steps away from the finished form. So you have to make sure that your definition of the starting material that you try to put into the process meets that requirement. Otherwise, you end up in trouble with the FDA. Okay, now to the nitty-gritty. You're going into the first clinical studies out of the R&D. What is actually expected for you to have done at this stage? The facility at that stage has to be clean, adequate space, and you have to make sure that you have contamination control in place. Either contamination by the materials that you bring in, or the environmental controls that, say, your equipment is clean, that your ventilation system doesn't bring in any adventitious agents, etc. For the process, you should have a written down procedure defining your process. The equipment needs to be adequately controlled, especially when it comes to cleaning and the contamination risk. The materials that you use should be fully documented. You should have specifications, draft specifications initially for the materials that you use in your process. They have to be suitable for the intended use, and you should at least have a certificate of analysis for each batch of material that you're using in the process. So suitable for intended use, as I mentioned before, try to use pharmacopoeial grade reagents wherever you can, because it makes it so much easier to register the product later on. Consistency batches at R&D scale. You need to have sufficient information, which you get by repeatedly producing your material at R&D scale, that your process is actually reliable and reproducible. So consistency batches would be deemed to be, say, for example, 3 consecutive batches of your production that deliver product that meets your draft specifications. If you have that assurance, then it is safe to start going into the clinic with the material. The analytics. Somebody earlier on made the comment that analytical methods should be validated. Well, I would say for phase 1, phase 0, you can't have completely validated methods because you haven't got reference standards and you don't know enough about the product yet. So at these early stages, you throw as many analytical procedures as you've got at your disposal at the product to try and gain product knowledge and understanding on what your product actually is and how it behaves. What you need to have are analytical methods that are documented, that are scientifically sound and reproducible. That means they have to be specific for what they're analyzing, they have to be sensitive enough, and they have to be accurate. So in order to meet these requirements, you also have to make sure that your analytical equipment is qualified and calibrated, at least. The method development at this stage in the transition from R&D to the clinic is running pretty much in parallel with your process development. For the product, it is important to make sure that the impurity and purity profile is equal to or better than the R&D batches that you have used in the toxicology studies. If you have any new impurities or changed impurity profile for the worse showing up in the commercial production, you're in trouble and have to do, as previous speakers mentioned, you have to do bridging studies and maybe repeat your tox studies. Of course, your safety profile from the tox studies isn't validated as soon as you have a different quality profile of the product in the early API manufacturing stages. You should establish draft specifications and again, only use a limited set of high confidence and reliable analytical methods for that. Importantly, you also have to consider that you set up stability studies as soon as you can. Remember, when you're doing the first in-human studies, you don't know how long the trial runs. If you know what your indication is going to be, make sure that the shelf life that you're assigning to the product is at least covering the intended clinical trial duration. One way to manage that is actually looking at accelerated stability studies by having, say for argument's sake, conditions where, say, if you store your product at 2 to 8 degrees, run the accelerated at 25 or maybe 30 degrees, it's unlikely to be stable at 40 degrees. But the accelerated studies will give you a heads up well in advance of when the crunch point comes that the product turns unstable. So Arrhenius principle, 10-degree increase in temperature means doubling of the reaction time. So if you run, say, 25-degree study is your intended use, 35-degree is your stability. That means you get a indication twice as fast whether or not the product is going to survive the duration of the study. And that applies throughout the development process. It was already mentioned that when it comes to sterile products, the PGOs and GMP requirements for sterility process and testing apply. The sterile products are just non-negotiable. You have to make sure that you meet those requirements. The batch of product we've already talked about, you have to make sure that you define exactly what a batch is, if it is a pooled batch or a single batches. You have to have a system in place which assigns batch numbers so that you have full traceability, and of course make sure that the batch records are accurately recorded with all the results from production and testing, and that those documents are also actually readily retrievable. Retention samples are important to keep. On the one hand, you need them for reanalysis if required. On the other hand, they're also very useful if you have a couple of old things sitting in the fridge somewhere or on the bench. And a year down the track, when you're in the phase 2 or phase 3, say, oh, well, you know, I mean, is my product good enough? Analyze one of the retention samples and you've got an indication whether or not the product was stable for this extended period of time. The real-time studies, especially for stability, I can't emphasize enough how important it is to keep that running as early as possible. Because if you say want to get stuck into a phase 3 study, haven't got a shelf life, well, if you want to have at least a 1-year shelf life, that means you're losing 1 year before you can actually go into the clinic, because the FDA will want to see what your shelf life actually is. And Catherine already went into the details on what is required for the product specification file. You should initiate that as soon as you get into phase 1. Additional activities when it comes to phase 2 studies. You have to manufacture the product in a GMP licensed facility. That's non-negotiable. The facility has to be fully qualified. As already mentioned, you have to have a calibration maintenance program running, and cleaning validation has to be in place for both the facility as well as the process. On the process side, by the time you're in phase 2, you're intending to improve the purity and impurity profile. So you're fiddling around with the process, trying to increase yield as well as the purity of your product, reduce the number of impurities wherever you can. But at the same time, you also have to be wary that in the scale-up process that you don't get new impurities or other products coming up which you haven't seen in the earlier studies, because they might invalidate your tox validity. Usually you run a significantly increased number of batches during phase 2, and you conduct less changes that you did in phase 1, and you're bedding down the process more, and you identify process controls. On the analytical side, David has already mentioned how important it is to have a reference standard identified. You should have at least a working standard established by the time you're in phase 2, and review the suitability of analytical methods. Whereas initially you just throw whatever you've got at product, characterize it. But now it's time to review all your results and decide, okay, which ones of the methods that I'm using are really key methods that help me characterize the product and ensure its reproducibility in the manufacturing process. You also need stability indicating methods. And for that, you need to know something about the degradation profile of your product. I'll get into that in a minute. The product characterization has to be conducted in an ongoing manner. So you're expected to have a more substantial understanding of the structure, the purity, and the impurities that are present. And at least at phase 2, you should start the analytical validation efforts and continue with that as you go. For the product, again, stability trials, set them up as early as you can, accelerate in real time with materials from the scaled up process, compare the results of the earlier studies to see whether or not there have been any changes in the stability pattern. You again would conduct another review of the available stability data to define preliminary retest or shelf life or stability period. You also need to identify what type of container closure system you use for the storage of your material. That is important because at a later stage in phase 3, you're expected to have information available to which extent any sort of primary packaging material interacts with your drug product or drug substance. When you're conducting stress studies to determine the degradation pathways, you have to look at thermolytic as well as hydrolytic effects, so base and alkali, and acid exposure, oxidative process, and photostability. As much as you know about the degradation pathway will help you to identify the search for impurities and making sure that you actually detect them with your suitable methods. The product specifications, again, need to be reviewed at that stage. By the time you get to phase 3, the process should be finalized. The synthesis, real and purification steps are bedded down and defined in detailed written master batch records. The in-process controls that you have identified by a pre-control point analysis earlier on must be defined and have acceptance criteria set. Process yields is another way of ensuring reproducibility of your product and your process. So you have to define, based on the production runs that you have conducted, what your expected process yield is. If the process yield changes for any reason, that's an indication that something in the process has not run the way that you expected. Scale up is another thing that you should consider at phase 3. On the one hand, you want to make sure that you have enough material available to satisfy the phase 3 trials. But if you're smart enough, because you're starting the validation at this stage as well, you're looking to manufacture the phase 3 material at a scale which allows you easy scale up to commercial. There is a rule that essentially a 5 to 10-fold scale up is accepted by regulators with minimal revalidation of the entire process. If you're ending up with a small quantity process that just satisfies the phase 3, you will not have a process that is viable to go into early commercial. I mean, you might have to do 10 or 20 production runs just to satisfy the launch of your product. But if you can scale it up by 10-fold, you can get away with 1 or 2 batches. Analytics, definitely in phase 3, the method validation has to be completed and the methods have to have this criteria established and the characterization should be completed. For the product, you again start putting material on stability trials from the finalized process. Again, accelerated and real-time studies need to be done for the stability and temperature cycling. With the accelerated, the ICH guidelines only ask you to run, for example, the 40-degree accelerator at 6 months. In my opinion, if the product is behaving and going well, run those as long as you can, because of the Arrhenius principle with the accelerated reaction time, you will always get advance notice if anything does go wrong with your product. So even though it is not a regulatory submission submissible evidence of your product stability when you have 12 months at 40 degrees, but it gives you the assurance that you at least can expect a 2 to 3 year shelf life of your product at 25 degrees when it is stable for a year at 40. And that applies all the way through whether you're going for freezing conditions versus fridge condition versus below 25 or 30 for the various conditions there. Temperature cycling, exposing your product to a freeze thaw cycle and then exposing to heat is important to conduct because it helps you assess deviations in the distribution chain later. I mean, you will always end up with scenarios where you ship your product around the world, and a container by chance gets left on the tarmac, either in winter by -20, -30 conditions, or it ends up in the Middle East at 40 or 50 degrees or 70 degrees on the tarmac. If you have that information on the temperature cycling, how your product is behaving, you can then make a ready assessment whether or not any product that has gone through an excursion in the transport chain is still fit for use and will meet its shelf life specifications. Material compatibility, I mentioned that already. Any sort of material that is in direct contact with your drug substance needs to be tested for material compatibility, and that goes for your packaging as well as for any materials that you use in your manufacturing process, like seals, stainless steel containers, hoses, whatever the product comes in contact with. You need to establish whether or not those materials impact in any way on your drug substance. The available stability data, again, need to be thoroughly reviewed, and a retest data stability period needs to be finalized. For stability studies, it's important that you actually, as I mentioned earlier, use the container closure system that you use for the real commercial product in a scaled down phase. So you want to make sure that your surface exposure as well as headspace in the stability study simulates what your final commercial container will look like. All impurities are expected to be fully identified and qualified at this stage, and product specifications should be finalized. Specifications, they need to be fit for use and need to be justified. You have to have acceptance criteria. Don't forget that specifications are method specific. So if you change your method, you also need to review your specification and see whether it still is appropriate for the type of method that you're using. The types of specifications that you require are, on the one hand, in-process specifications, which help you ensure that the process is running according to plan. And then you're dealing with release and stability. The expiry specifications usually apply at the end of its product shelf life on stability, but it is always worthwhile, given that the products will deteriorate with time. You want to make sure that a product that has just been manufactured is not close to the expiry spec. So you actually would set up a set of release specifications, which is much tighter than the product specs that apply at expiry. And you will either register them both as release spec versus expiry spec. But again, consider that the acceptance criteria that you set are tight enough. If they're too wide, the regulators will say, okay, sorry, no go back, and we want, say for argument's sake, if a pH that you list for your product might be pH 5.6, your process delivers from 5.0 to 6.7 or whatever, and you set a spec from 3 to 10. Sorry, doesn't cut it with a regulator, because they say your process is much tighter. We want to see the specs much tighter to reflect what the actual process capability is. The product characterization specifications are one thing where you actually define what your product is. In addition, you also have to consider the pharmacopoeia requirements. That would be, for example, impurities like endotoxin, sterility, osmolality, etc., depending on the intended use of your product. But that actually goes into the drug product specs more than for the API when I talked about osmolarity. The specs are different depending on the intended route of administration. As you all understand, an injectable has much tighter specifications when it comes to impurities compared to an oral or a topical one. And then you have to deal with sterile versus non-sterile products. Typically, you have to describe your API, whether it's with a solid, liquid, color, etc. You have to have at least 1 or 2 identification tests, which are specific for the active ingredient. Assay or quantity, again, has to be specific for the API and has to be stability indicating, so that any degradation that may happen is being picked up by changes in these parameters. The impurities that you're dealing with are either organic or inorganic, or from a microbial perspective. They may be originating from the process itself with the material inputs that you're using, or processing aids that you use in the process. Or they could be product related, could be related substances or degradation products. You have to have specifications for each of them, and depending on your qualification, set the limits for acceptance at the appropriate levels. When it comes to physical chemical properties, for chemicals that are usually in solid form, it's a particle size, the polymorphics that you have to look at, whether or not you've got a chiral compound. For biologicals, it'll be the biological activity, potency, heterogeneity pattern, whether or not you've got post-translation modification, whether you've got methylation on RNA-based products, etc. So at the end, product safety and efficacy comes back to the fore again. The data, keep in mind that the data generated in the clinical trials are only valid for the drug substance that you have manufactured and analyzed by the documented and validated manufacturing process. That defines exactly what your drug substance is. You have tested it in the clinic, you have tested it in the tox. That is a safety profile. If you make any changes, you then have to demonstrate that these changes do not change the safety and efficacy profile of the API. So you haven't got any new impurities coming. I mean, there's plenty of examples where a manufacturing process has been changed, and was it on the famous Shodenko in the late 80s was a case that an impurity in the level of less than 0.1% showed up, which wasn't picked up initially. But when the new product came to market, I think about 180 people or so died because of that impurity. So you can never take anything for granted. And that's it for me. Thanks very much.
Thank you so much for that, Jürgen. I'm sorry, we're going to just hold questions until after the next speaker, if that's okay. For those of you joining us online, if you do have questions for any of the speakers, please feel free to put them in the chat. There will be an opportunity to ask those afterwards. I do just want to make a quick observation in response to Jürgen's point really early on in his talk, that the purpose of all of the stuff that we're talking about today is at some point to develop a commercially viable product. To do that, at some stage, there's going to be some level of probably licensing with a big pharma company. And when your business model is to license to a big pharma company, as Rob McGuinness, a local IP and commercial lawyer, puts it, it's not about the risk that you're willing to accept. It's about the risk that your licensee is willing to accept. So all of this work is about making your product more commercially attractive as well, or not shooting yourself in the foot. Our next speaker now is Craig Rogers. Craig is a registered pharmacist and toxicologist and holds a law degree. He's had 30 plus years of experience in the pharmaceutical industry, initially holding senior roles in regulatory affairs and scientific affairs at GSK, Faulding, AMRAD and IDT. More recently, Craig was the General Manager and Chief Operating Officer and Chief Executive Officer of the Early Phase Clinical Trials Unit CMAX and Nucleus Network, where he was responsible for the execution of 300 plus clinical trials, including many first-time in human trials. From his time in these roles, Craig appreciated the difficulty in finding appropriate facilities for investigational product manufacturing for clinical trials. This resulted in him establishing Pharmaceutical Packaging Professionals Proprietary Limited, PPP, in 2008, a specialist manufacturer of investigational medicinal products. Craig was CEO and General Manager of this facility for 10 years before overseeing the manufacture of around 6,000 batches of IMP for trials in Australia and overseas, and employed approximately 80 full-time staff. In 2018, Craig managed the sale of PPP to PCI Pharma Services. Earlier this year, in response to many previous clients' requests, Craig established Sintro Proprietary Limited, a new TGA licensed GMP facility for investigational product manufacture, warehousing, distribution, and direct-to-patient pharmacy services. Over to you, Craig.
Thanks very much. Just confirming you can hear me okay?
We can, yes.
Yes, great. So look, my presentation is going to be more around the practicalities of finished product manufacturing. As we've heard from Catherine and others, that the client who engages the finished product manufacturer for a clinical trial takes on the GMP responsibility or GMP responsibility of the quality of the product. So following my experience over a number of years working in this space, I've tried to focus on the key issues that need to be addressed by that client in being comfortable that the product meets GMP requirements, and when they sign that CTN form, that they're understanding the responsibilities they're taking on from a GMP perspective. I'll focus on these 7 points, though it's true to say that a number of them have been commented on in the previous presentations. So I'll only briefly reiterate them, but I think it just underscores the importance of a number of these issues that come up time and time again when engaging GMP manufacturers and when trying to manage your investigational product manufacture. As we've heard, phase 0 and 1 trials are not subject to the facilities that manufacture the finished product for those studies are not subject to GMP licensing. However, I think I would concur with the other speakers that you need to understand that that the GMP rules are in place, often to protect the safety of the participants and ensure the quality of the product. So I think it's generally good practice to use the GMP facilities for these activities. I think also it's a risk-based approach. When you look at the cost, for example, of doing drug in capsule for a phase 1 SAD/MAD, it may come to, for example, 100,000 by the time all the analytical and the capsule filling is completed on a per cohort basis. But the reality is you're investing 2.5 to 3 million potentially in your clinical trial. So in the scheme of things, to risk going into a facility that's not familiar with GMP requirements is probably too great for the cost of the clinical trial that you're embarking on. Phase 2 onwards, definitely regulated by the TGA and subject to GMP licensing. And again, the PICS guidance for chemical entities or finished products, Annex 13 describes those requirements. So what are some of the facility issues that you should need to consider when engaging a GMP manufacturer? Well, I think as, again, Catherine alluded to, in the license, it needs to be specific to the formulation that you're manufacturing. So if you're running coated tablets, the license needs to confirm that facility can make coated tablets. Secondly, it needs to specify that they're authorised to make investigational products. Investigational products come with additional risks and additional licensing requirements around the audit process, in particular around randomisation or label control, for example. So that's why it's important that an investigational product is included on the licence. And I think importantly, one point that hasn't been mentioned is that it should include a release for supply step on that licence. Because a company can make a finished product coated tablet, if there's not a specific line item that says release for supply, in other words, the facility can undertake release of supply, and there's an authorised person, then they're not in a position to release that product for clinical trial use, and you may have to engage a third party to do that. But it's probably safest to make sure that the licence holder who's undertaking or manufacturer has the appropriately trained authorised person who is listed on the licence to fulfil that obligation. Aside from the GMP licensing, the next step is really establishing a GMP agreement or a technical agreement and a service agreement. The service agreement really specifies the activities that are going to be undertaken, the costs, confidentiality, indemnities, those types of things, whereas the GMP agreement is really the step-by-step list of undertakings that you will take and the GMP manufacturer will take, and assigns the responsibilities for those activities. So important things are obviously the release for supply, who's going to undertake that, recalls, issues around warehousing, distribution, stability studies, and who will make judgment on shelf life extensions, for example. So normally that document is certainly separate from the service agreement, and that is the preferred approach. And it's generally a 2 to 3 page tick box document outlining client and manufacturer's responsibilities. So in terms of the manufacturing process, investigational product manufacture does throw up a number of difficulties beyond standard commercial manufacture. One of those specific difficulties is sourcing of raw materials, packing materials, excipients that might be used in the manufacture. There's a number of reasons for that. Identifying suppliers can be very difficult for excipients. In particular, if you're looking for suppliers that are compendial grade, have a good track record in the supply of these materials for manufacturing of pharmaceuticals. Often they have minimum order quantities and have very long lead times. The other aspect is when you're buying 100 grams of material for a clinical trial and then asking a supplier who you need to qualify as part of the GMP process, please fill out a 15-page document and we may want to audit you. Generally, the response isn't positive. So you have to be really mindful of the suppliers you're selecting, and often the more, a judgment of how robust and how good a quality a supplier is, they often have their own qualification documents that they can supply you as a package. And that's normally a good judge of whether they're going to be a good partner to work with. In addition to that, there's release testing of all the materials you're receiving. So you have to determine the extent, the qualification of the methods, and the suitability of those products for human use. So it is definitely one of the biggest issues we often face in the manufacturing sense, is identifying appropriate suppliers of excipients, packaging materials. The API sense tends to be less of an issue because the client tends to supply the API or is engaged with someone who's manufactured the API. I think we've heard from another number of people about the manufacturing practice records. Really important, they're clear, unambiguous, and absolutely err on the side of collecting more information than less. The reason for this is that you don't have process validation. This data is leading up to helping you develop your process validation for your product. So collecting information about mixing times, amount of pH adjusting solutions you're adding, the frequency you have to add them, mixing speeds, temperatures, those types of things will provide you a lot of information for the next batch. And I think also if there is a stability issue, sometimes it becomes a way to look back at that batch manufacturer and maybe identify where that stability concern may have arisen from. I think the other important thing is I think you should always manufacture, where possible, placebo first to avoid cross-contamination with the active. Cross-contamination is a big issue in clinical trials, in particular between active and placebo. And also, if it's an early phase study, you don't have a lot of safety data on the active, so you don't know what safety levels are appropriate. So cross-contamination is a difficult issue in clinical trials and one that needs to be managed. We certainly would normally recommend, where possible, and in particular for small-scale manufacture, dedicated equipment for active and placebo, and that equipment is stored and cleaned separately. The reason for this, it does avoid a lot of the costly cleaning validation that's very expensive when you're cleaning equipment. It still doesn't negate the need for obviously cleaning of your facility and understanding what requirements are going to be needed there, but certainly dedicated equipment for active and placebo is highly recommended. Another point is comparator selection in manufacture. So we engage with a lot of universities and undertake a lot of research-based studies, and a lot of these studies are phase 3, phase 4, looking at existing marketed products versus placebo. In these studies, we're purchasing the active off the market, but there's some really important considerations if you're trying to make a placebo to match that. So the first thing I would always suggest, if you're going against an active tablet, try and avoid one that doesn't have a colour on it. It's much easier to make white tablets to match the placebo and match the active. Avoid too much livery on the tablet if possible. We find in particular with the big pharma that they have arrangements with the tooling manufacturers that prevent them making a matching tooling set. There are ways around that, but the reality is it does complicate the process. Look for a product with the longest shelf life. You can find that on the ARTG, so it'll have the registered shelf life, and then either speak with the suppliers or, if possible, the manufacturer to see how close a batch is being manufactured to its release onto the market. So often they'll be 6 or 12 months before it even gets to the market, so that could consume a lot of the shelf life. But you just need to be cognizant of that because you don't want to be doing continuous shelf life extensions or having to remanufacture. The other important part around the comparator selection is we find, in particular in the generic space, where a lot of the comparators are bought as generics, the market can be a little bit fickle depending on the cost of the generic and the reimbursement that's happening and the competition, and some products come on and off the market, and that can be quite disastrous in a clinical trial where you're doing it in tranches and you may be buying another tranche a year later, and then or all of a sudden your placebo doesn't match the active product on the market. And I can assure you that's certainly happened on a number of occasions. The other part is, will the study involve overseas sites? So really discussing with the sponsor what the likely trial sites are going to be, because that really has a big impact on your comparative sourcing as well, because it can make your regulatory process very difficult if you're active from a market that doesn't have a mutual recognition agreement with, for example, if the study's being done in Europe, and that means you won't be able to generate a regulatory package that will be suitable for the clinical trial in that country. So understanding where the comparator is going to be used is really important up front, because that will also dictate where you'll buy it from. So, for example, often we have European arms in studies. We'll tend to try and buy the drug from Europe rather than sourcing from Australia or other territories, just because it makes the regulatory process a lot simpler in the end. Some of the product considerations. So I think there was some discussion about phase 1 studies, and having been at Nucleus Network for about 11 or 12 years and CMAX for 4 years prior to that, having done hundreds of first-in-human studies, I can assure you the majority of oral studies, topical studies, and injectables would go in with a fairly simple formulation. So drug in capsule only is probably the most common formulation you would see. There's a number of reasons for that, and this is not just a small pharma or biotech issue. We had multiple preferred provider agreements with big pharma, and it's really about failing fast for them. It's about getting PK data that is commensurate with the toxicokinetic data, and looking for unexpected safety signals. But probably more recently is building in PD assays in the healthy volunteer studies and trying to get a PD assay that will dictate your likely dose in the phase 2 studies. The other reason they don't come in probably with anything really close to the final formulation is because the T-Genero issue was mentioned recently. That has caused a significant reduction in the initial starting doses. So it's really difficult to understand where you're going to end up with. You might be 1,000 times below the likely clinical dose for the first dose. So having a formulation that's going to cover that whole range from an oral point of view is pretty difficult. The other considerations for over-encapsulation, so we often do tablets and over-encapsulate them to blind. That's the simplest method. Obviously, it has some inherent problems. People can open capsules and see what they're on, and there are potentially some stability issues, but most of it's around the unblinding potential. The other issue is, how big is the tablet that you're going to over-encapsulation. If it's metformin, a 1 gram tablet, it's about 1.5 grams in weight. Over-encapsulating that will make something that would be very difficult to swallow. So you really got to understand the tablet you're talking about. Then there's the different types of capsules. The DB capsule is the clinical trial capsule that has a much wider body, and so you've got a lot more tablets into it, and the body pushes right into the cap, so it's very difficult to open. And then there's the classic Coney snaps. Again, understanding where your study's going. We use a halal capsule if you've got Middle East or sites that may require halal capsules. Colour, so is the tablet you're putting in it very bright, then you need to use a dark capsule that they can't see through. What type of rattle, what material is used to stop the rattle, such as microcrystalline cellulose to backfill the capsules to stop it rattling. And then the capsule type. So if the tablet has some is hygroscopic, a lot of these statins are, for example, then probably go for a HPMC capsule where it's got a low water content versus a gelatin capsule which has 5 or 6% water in it and cause stability issues or disintegration issues for the tablet that's inside. Normal development time for over-encapsulation is 1 month to less. There's often a lot of published data that can help support an over-encapsulation. Other options are powder in capsule. So for phase 1, we do a lot of drug in capsule only, and we just escalate on a per cohort basis. Again, you need to have some understanding of the API, its bulk density, is it hygroscopic, colour, moisture content, just in terms of handling that API when filling capsules. The matching placebo, bulk density is very important. So what happens if the API is very heavy and you use MCC, which is very light? When you fill 30 capsules in a bottle, you can discern maybe a 15 or 20 gram difference in the bottle weights and can unblind the study. So you need to understand the type of API you're using in the capsule. Often with powder and capsule, there may be some blend work required. And certainly when you get to phase 2, you're often doing blends because you can fill a lot more capsules a lot more quickly. But then you've got excipient selection and some development work around that. The other aspect is analytical is very expensive at phase 1 relative to the actual manufacturing costs. Often it outweighs it depending on the extent of analytical stability you're doing. So, you know, if the fill weight is greater than 75% of the API in the capsule, then you can normally do uniformity of weight as opposed to uniformity content by assay. That can certainly reduce costs and timelines. And then obviously in powder and capsule, there's method development issues with dissolution versus disintegration, and dissolution testing and development is quite expensive, especially if you're using a variety of medias and looking at something that's is able to discern changes in formulation characteristics. Then when it comes to tablets, we don't see a lot of tablet development certainly in our facility and in so much in Australia. It tends to be done at phase 2 and phase 3, and maybe a big farm has license at that time. But there's certainly a lot more development work required. There's excipient stability and development, a lot more method development, in particular discriminative dissolution methods, R&D stability requirements, and certainly increased API needs for tableting. So manufacturing process from a sterile point of view, there are different considerations for an injectable. There's excipient selection. Are they generally safe? Are they excipients that you use broadly in products? Your container closure selection, do you need amber? What type of rubber stopper? Does it need to be something that's the product's reactive? So you need a Teflon coated stopper versus a bromobutyl stopper. Stability in the container, for example. Can you match the pH, match the placebo, especially if the drug's coloured? If it's freeze-dried, it's even harder. Can you match the pH, the osmolarity, etc.? And then there's formulation considerations. Are you giving the product as an IV push? Most first-in-humans are IV pushes from the product. It's rare that there's so much of a reconstitution into a bag. So you've got to consider things like pH, what volume are you giving, and is the pH an issue? Osmolarity, and then filter compatibilities if you are putting it through a filter, for example, both in the manufacturing process but also during use of the product in the trial. And then obviously there's the standard issues around sterility, endotoxin, particulates that differ to a non-sterile product, and then shelf life setting and in-use stability requirements in terms of how do you reconstitute in the syringe at the pharmacy, how long can it stay at 25 degrees, what plastic lines is it compatible with. So you need to do a number of in-use stability considerations when you have an injectable product. Labelling, probably one of the biggest issues in clinical trial. So label proofs, there's obviously TGA requirements for labels. Again, as I said previously, understanding what region the product's going to can mean you can make a combined label, which provides you a lot more flexibility in your stock. So if a country's not recruiting, you don't have to send that stock to them. You can use it in other territories. So normally you can combine New Zealand, EU, you can combine a lot of the Southeast Asian countries will take English labels, in particular if it's a hospital product. The other big consideration, and probably 50% of most GMP manufacturing sites work in investigational product, is shelf life extensions. So we would absolutely, reiterate that you should try and get your retest expiry date in a position that you can over-label it if you have to. So don't have it in the middle of all the text. If you've got a small container inside, like less than a 2 ml vial or ampule, don't have a shelf life on that container, only have it on the secondary container. So thinking about those things in advance, we're going to make it easy to do shelf life extensions. Randomization list control is critical. 100% double checking the labels, 100% double checking against a unchangeable randomization list. So receiving a PDF copy is a minimum, and then potentially a CSV file if you have to mail merge when you're doing long production runs with lots of randomization numbers. Who will store, who will have access to it are key issues that the sponsor should be concerned with, and I know that, for example, the TGA would be concerned with as well. Label type, there's direct thermal and there's thermal transfer. Thermal transfer tends to be a better label. It tends to be better with a rub test. So if you've got products rubbing up against each other, you need to consider the type of label you're using, the type of printer. And then again, if you're storing it at room temperature 2 to 8 or -20, paper labels will without doubt crimple at -20, condensation gets under them. Cartons will deteriorate. So you really need to think about the carton selection and the label selection based on how you're storing your product. Again, for carton type for secondary packaging, there's not many hospitals that would accept just vials. So you're normally going to need a secondary carton. So you talk about the carton type, thickness, the tamper evidence, and the integrity, as I said, when it's stored at room temperature, refrigerated, or in many cases, -20. Specifications. Normally you should use compendial specification if it's one described for it. There are specific TGO monographs as well for tablets, capsules, and injectables that need to be followed. The specification should be agreed upfront and signed off by the client and the manufacturer. So in terms of documents the client would see, they'd be the manufacturing batch record, the specifications, and the label approval form, which tend to be the 3 core documents after they've signed the GMP agreement. So in terms of the validation, normally at phase 1 and 2, you've at least validated the method for the assay. Impurities might be still by relative retention time. Certainly by phase 3, you should have stability indicating methods. For placebo, you need to also consider what testing you're doing for that. Obviously, it's sterile, which needs the same sterility, endotoxins and particulates. But the other part is often we would do absence of active, so whether it's an FTIR or HPLC identification test, just to show that there's been no cross-contamination. I think Catherine reiterated this, and this is probably one of the big TGA findings, is the lack of a finished product specification file. So it's really important that the manufacturing company you're working with is developing a specification file, has SOPs to describe that, and produces that. And that's for batch n = 1 through to batch n = 100. So if you're doing a one-off batch, you still need to develop a specification file. Obviously, the information in that may not be at the same level as a 100 batch, repeated manufacture, but the fact of the matter is you definitely need a specification file. Stability, probably the bugbear of most clinical trials, and the thing that results in studies stopping, studies halting for a period of time, or additional work in terms of remanufacture or shelf life extensions. So stability is a GMP activity, needs to be undertaken by a GMP licensed facility. Often we find that stability is not quite thought about upfront, and what happens is they turn around and say, oh, yes, we need stability, but they haven't got a reference standard and the columns have long lead times, so automatically you've got some delays in the project startup. Methods should be validated, as I said, commensurate with the stage of development, and that may be just recording impurities by relative retention time in an assay at phase 2 through to actually identifying impurities in phase 3. Storage should be in use with clinical trial conditions, and really important to do accelerated stability because that gives you to 2x to 4x real-time data, and so it gives you some runway on setting a shelf life that you can then corroborate with real-time data. As I said, in-use stability should also be conducted for injectables, so syringe and infusion line compatibility, in-use stability. So as Judy indicated, she had a 30-vol product she had to make up. That would have taken, the pharmacist probably a couple of hours to reconstitute. By the time they got everything up and set up, is the product refrigerated and it was okay to stand out of there for 2 hours while they're reconstituting. So those types of things are really important at phase 1 and phase 2 when you're making a product. Shelf life extensions have to be planned. There are minimum data requirements. You have to understand what are going to be the minimum data requirements to extend the shelf life. Who will prepare and sign off on the memo to the manufacturing site to authorise the shelf life extension, and who will conduct the shelf life extensions. It should be noted that shelf life extensions at site can be undertaken by the site and don't have to be done in a GMP licensed facility. So really up to the sponsor and where their product is as to whether they're going to initiate extensions at sites. However, in those instances, we would write, summary batch records, we prepare the labels, they tend to be in a standard design, and then send that to the site so they can execute that process. Of course, there needs to also be contingency if the shelf life extension fails, or the shelf life fails. So that's ideally why you should start your stability on R&D batch. Well, it's not always the case. It will give you some accelerated data in advance and help you predict any potential failures. Monitor real-time stability against the predicted extrapolated data. Make sure you're on track. And if necessary, you need to be in a position where you can undertake remanufacture if you have to, if there's a significant failure in the stability of the product. You should also need to consider whether you place your placebo on stability. EU in the CTA applications invariably requires your placebo on stability. It tends to be a very limited program, so 0, 6, 12, 18, 24 months tends to satisfy them, but it's something you should proactively do, in particular if you're having your product used in the EU, for example. So finally, on quality requirements, the manufacturer really should have a dedicated product group independent of manufacture for quality, and certainly if they're audited by the TGA or any regulatory authority, that would be the case. They should have experience in reviewing clinical trial batch records, because they do have significant differences to just routine manufacturing, especially around blinding, randomisation, making sure the right randomisation list has been used. Those types of things are just critical. That quality group should undertake an independent check of the randomised labels against the list, and they should also perform line clearances between manufacturer, in particular to minimise the risk of active and placebo mix-up that may occur in manufacture. Increasing, and the quality group should be proactive, increasing the in-process controls within the batch record to provide some assurance around the manufacturing process, but also to provide that information for process validation and future batches and product tech transfer. Make sure that the quality person is an authorised person, is appropriately trained for investigational product release, and that person is on the TGA licence, authorised to undertake that role. They should also ensure the records are retained, including randomisation lists, finished product samples, and that should be consistent with the GMP and technical agreement. And if they're asked, and we are regularly asked to give the executed batch record to the sponsor to make sure that the quality person checks that, firstly, that person, if they're blinded, you de-identify the information and get it to them, and confirm if they're unblinded, and confirm that in writing before you would give them an executed batch record that may potentially unblind the study. In small biotechs, in most cases, in a lot of cases, should I say, the quality person is still blinded to the study, so we would often just send through de-identified batch records that would take out randomisation lists and randomisation numbers. So the other part the quality person should be is managing facility audits, looking at third-party audits of vendors you may be using, and also participating in QP audits if the product is going to go overseas into Europe, for example. In terms of the other important thing in clinical trials is recalls. So the site should have a mock recall procedure. They should be familiar with the processes with TGA liaison and sponsor liaison in terms of agreed time frames. And if required, and often we were asked, to provide a 1-800 number to undertake emergency unblinding and understand the process by which that would be done, and for that to be explained in the technical agreement. So I try to whiz through that because I knew we were a bit short on time. So happy to answer any questions that you might have.
Thanks very much for that, Craig. And we have had a colossal amount of information this afternoon, and just some amazing experience that's been shared with us. So I just want you all to join me in thanking all of our speakers one more time: Judy, Catherine, David, Paul, Jürgen, and Craig. Thanks. Now, we do have some time for some questions, and I would ask those of you who are online, if you have any, please do pop them into the chat. Hopefully some of our speakers will hang around for the drinks afterwards, and so people in the audience will be able to ask them. Do we have any questions? While people are having a bit of a think about that, I think that there's sort of two really, really clear messages that came through from all of the speakers. The first is, it's not too early to think about that. And that's just a different way of saying begin with the end in mind. But often when I'm speaking to early stage companies or academics who are thinking about spinning something out and commercializing it, they say, oh, look, I don't need to worry about scale up. It's too early to think about that. I don't need to think about the clinical trials. It's too early to think about that. But I think that our speakers today have really made it clear it's not. Think about it now, think about it early stage, and don't waste the time. The other one that really came through strongly, I think, was just because you can doesn't mean you should. And I think that's in respect to phase 0 and phase 1 trials. And maybe you can do a trial with non-GMP material, but really, as all of our speakers have shown today, you shouldn't for lots and lots of reasons. We don't have any questions coming through on the chat. Does anyone in the audience want to ask any questions, or should we adjourn early and talk to our speakers outside? It looks like we're going to adjourn earlier. Before we go, I will just pass the microphone to David to say a few closing words.
Good. Thanks very much indeed, Duncan. I can't really remember a seminar over the times I've been organising these things, and the farm group's been organising them, when we've had so much information collated into an afternoon. It's really been terrific. And also, I would also suggest that there have been 3 or 4 points, which I won't reiterate, that have been reinforced independently by many of the presenters. We haven't really talked to each other to the extent of say, well, we all should say this about stability and the like. We've all come up with this independently. So I'd like to think that these sort of reappearing messages are taken on board by the folk who are involved in the early drug development. I'd like to thank very much the Pharmacy School for providing the venue today, to Ab Initio for the catering, to Niall Wheat in particular, if you could pass on to your colleagues in pharmacy, Niall, in terms of organising the setup and the like, and you know, we're very, very grateful for you. I'd like to thank also, of course, the chair and presenters this afternoon. It's very apparent that a lot of preparation has gone into these talks. They just don't happen. And we thank you very much for all the time and effort that you've put into this. So I'd also make a quick commercial to the effect that the next farm group webinar seminar is as near as the 25th of October, and which will be on data integrity. Details have been circulated already, and all I can suggest is that it should be a good seminar, and we look forward to your company there as well. So again, thank you very much for attending today, and we hope that the farm group can continue to present these sorts of events.